all living things, from the smallest single-celled organism drifting quietly in a pond to the largest animal moving across the land, share a profound and unifying feature. Each is built from cells, those minute units that together create the intricate fabric of life. If you imagine looking closer and closer at the structure of a living creature, peeling away tissues and organs, and even individual fibers, eventually you would arrive at these microscopic rooms of life. A cell is a space enclosed by a delicate boundary, filled with the machinery that sustains existence, and within it, the secrets of growth, repair, and reproduction are carried out in silence. The ability of cells to divide is one of the most remarkable processes in all of nature. It is quiet, patient, and precise. When we think of life continuing, we often picture seeds sprouting or infants growing, but at the most fundamental level, it is the simple act of a cell dividing into two that makes continuity possible. Each division is like a soft rhythm in the background of existence, repeating over and over again, unnoticed, yet essential. This rhythm has been ongoing for billions of years, stretching back to the earliest forms of life that once floated in ancient seas. Those first primitive cells learned to copy themselves, and from that ability, every plant, every tree, every animal, and every human being has come to exist. The story of cell division is not only one of reproduction but also one of maintenance. Our bodies, for instance, are not static sculptures made once and left unchanged. They are dynamic, living tapestries, constantly being renewed by the quiet labor of their cells. When skin is cut, the wound does not remain open forever. Slowly, gently, cells around the edges of the wound begin to divide, filling in the gap until the surface is whole again. When bones are broken, the same process helps knit them back together. Even the lining of the stomach, exposed daily to harsh conditions, is replaced again and again through the tireless division of cells. It is almost soothing to think that beneath every conscious action, walking, speaking, or even drifting into sleep, there is a hidden world of patient work being carried out. Cells multiply, ensuring that tissues remain healthy and that organs continue to function. This process is so reliable and so constant that we seldom notice it. Yet without it, no creature could survive for long. The quiet pulse of cellular renewal is what makes life resilient. If we follow this thought further, it becomes clear that cell division is not merely a biological mechanism, but a rhythm upon which the stability of life rests. Growth from childhood to adulthood would be impossible without countless rounds of cell division. The simplest injuries would remain unhealed. Even the immune system, which protects us from infections, depends on the rapid multiplication of specialized white blood cells. Each time the body detects an unwelcome invader, certain cells begin dividing quickly, producing armies of defenders to restore balance. And yet, though this process is continuous, it is not uncontrolled. Nature, in its wisdom, has built careful timing into the cycle of cell division. It is as though each cell carries an inner clock, allowing it to pause, prepare, and check its progress before moving to the next stage. This cycle of growth, preparation, and division has been shaped over vast stretches of evolutionary time, refined so that life may be preserved with the greatest fidelity possible. It is a cycle both delicate and durable, able to withstand small errors and correct them, but also flexible enough to adapt to changing needs. To fully appreciate the wonder of this, one might pause to consider scale. A single human body is made of trillions of cells. Every moment, millions of them are dividing, while others are resting in a quiet interlude, and still others are preparing for the next round. Extraordinary, yet it unfolds without noise or hurry, an immense orchestra of microscopic players performing in silence. Cells in the liver know when to divide, cells in the skin know when to pause, and cells in the bone marrow know when to accelerate, each responding to subtle cues. This organization allows us to live in relative harmony, seldom aware of the continuous renewal occurring within. It is tempting to imagine this activity as something hurried or chaotic, but in truth it resembles more a patient cycle of seasons. Just as spring follows winter and summer yields to autumn, the phases of the cell cycle move steadily forward, repeating endlessly, guided by invisible signals. Within each cell, DNA, that long molecule of inheritance, is carefully copied and distributed, ensuring that each new cell carries the full instructions for life. It is a fragile process, yet protected by multiple layers of safeguards, as though nature has wrapped it in care to preserve its continuity. This gentle persistence of cell division connects us with all living things. The fern unfolding in a shaded forest, the bird tending its nest, the fish gliding through water, all are sustained by the same unseen rhythm. It is one of the most profound examples of unity in biology, a shared pattern that crosses every branch of the tree of life. No matter how different species may appear, beneath their surfaces, their cells are following the same patient cycle. When you reflect on this, a sense of calm appreciation arises. The continuation of life does not depend on grand gestures or sudden movements, but on this quiet repetition at the cellular level. Each heartbeat, each breath, is accompanied by countless divisions within us, tiny acts of renewal that together create the vast and complex experience of being alive. The more one considers it, the more extraordinary it seems, and yet it is so familiar, so constant, that we often take it for granted. There is a certain comfort in knowing that, even as we rest, even as we close our eyes in sleep, this process continues without interruption. Cells divide, tissues repair, and the body renews itself. It is an ancient rhythm, one that began long before us and will continue long after. To lie still and breathe slowly while imagining this hidden cycle is to feel connected to the deeper fabric of life, to sense the continuity that flows through every living thing. As we begin this exploration of the cell cycle and the many ways it sustains existence, it is helpful to carry with us this image of quiet persistence. What follows will move into details: the stages of mitosis, the subtle differences of meiosis, the careful checkpoints, the intricate repairs, and even the troubles that arise when balance is lost. But for now, it is enough to rest with the thought that life endures because cells divide, softly and steadily, carrying forward the story of existence one cycle at a time. 2. The nature of the cell cycle. The life of a cell, though hidden from our eyes, follows a gentle rhythm, a cycle that repeats itself with quiet precision. Just as the days unfold into nights and the seasons move from spring to winter, so too does a cell pass through phases of activity and rest, preparation and division. This repeating journey is called the cell cycle, and it lies at the very heart of biology. It is through the cell cycle that living beings grow, repair themselves, and pass their essence from one generation to the next. Without it, the story of life would be brief and fragmented. With it, life is sustained in a continual flow. To imagine the cell cycle is to think of a circular path, one without a fixed beginning or end, always moving forward yet always repeating. Along this path are distinct stretches of activity, each with its own purpose. Some phases are devoted to growth, others to careful preparation, and still others to the act of dividing into new cells. It is not a hurried journey, but rather a patient one, shaped by countless generations of evolution to be steady and reliable. Most of a cell's time is spent not in division itself, but in what is called interphase. This is a period of quiet work, in which the cell lives its everyday life, growing, carrying out its tasks, and preparing for the future. During interphase, the cell does not appear to be doing anything extraordinary. Under a microscope, its nucleus remains intact, and its DNA lies in a relaxed, thread-like form. Yet, Beneath this calm exterior, the cell is hard at work. It is absorbing nutrients, producing proteins, and in many cases, copying its genetic material in preparation for the next division. Interphase is not a pause, but a long, essential prelude, much like the slow gathering of energy before a leap. This interphase is divided into three distinct stages, which flow into one another without abrupt boundaries. The first is known as G1, or the first gap phase. Here, the cell increases in size, builds up reserves, and carries out its specialized functions. A liver cell might produce enzymes to aid digestion, while a nerve cell might transmit signals. Each cell type performs its role while also quietly checking whether conditions are favorable to proceed further. Then comes the S phase, or synthesis, when DNA replication takes place. During this stage, the long strands of genetic material are carefully copied so that each new cell will have a complete set of instructions. Finally, the cell enters G2, the second gap phase, where it continues to grow and makes final preparations before division. This includes producing proteins that will be used to separate the DNA and form the structures needed for the process ahead. Following interphase, the cell enters the shorter but striking stage of mitosis, or sometimes meiosis, depending on the purpose of division. In mitosis, the goal is to create two cells that are identical, each with the same genetic information as the parent cell. In meiosis, which is reserved for the formation of gametes such as sperm and egg cells, the purpose is different: to reduce the chromosome number by half, allowing for genetic diversity when gametes merge. Though their outcomes differ, both forms of division are orchestrated with exquisite care. The cell cycle is often described as being under tight control, and this is true in a very literal sense. At certain points along the journey, known as checkpoints, the cell pauses to verify that everything is in order. Has the DNA been copied correctly? Are there enough nutrients and energy available? Is the environment safe for division? If the answers are not satisfactory, the cell may delay its progress, attempt repairs, or even enter a quiet resting state called G0, where it withdraws from the cycle entirely. This resting state can last for days, years, or even the entire life of the organism, as in certain nerve cells that rarely if ever divide again once they are formed. In this way, the cycle is not just a mindless repetition but a carefully monitored process. Each cell acts almost as though it possesses a sense of timing and judgment, able to decide when to proceed and when to pause. This vigilance is what allows life to persist with such fidelity. If cells rush through division without these safeguards, errors would accumulate quickly, threatening the survival of the organism. Instead, the cycle unfolds with a rhythm both flexible and precise, able to adjust to circumstances while maintaining balance. It is worth reflecting on how much of life depends upon this unseen cycle. Consider a child growing taller year after year. Beneath the surface, bones elongate, muscles expand, and skin stretches to cover the new form. All of this is made possible by countless rounds of the cell cycle. Each tissue depends on the cycle to increase its numbers, adding layer upon layer until the child becomes an adult. Even after growth slows, the cycle does not cease. Cells continue to renew themselves, ensuring that tissues do not wear away with use. In the lining of the intestines, for example, cells divide constantly to replace those shed each day. In the skin, new cells rise to the surface, as older ones are sloughed away. The cell cycle is the hidden engine of continuity, maintaining what might otherwise crumble. Plants, too, rely on this cycle for their silent growth. In the tips of roots and shoots, cells divide repeatedly, pushing the plant upward toward the sun or downward into the soil. Each leaf, each petal, each grain of pollen is born of these divisions. Even the rings of a tree, which mark the passage of years, are the result of cycles of growth and division layered one upon another. The quiet forest, the sprawling meadow, and the solitary flower in a garden all share this rhythm. In simpler organisms, the cycle may appear even more fundamental. A bacterium, with no nucleus and far less internal complexity, still divides in a process resembling the cycle's essence. It grows, copies its genetic material, and then splits into two. This simple repetition, carried out billions of times across the planet every day, sustains life in its most abundant forms. Whether in the depths of the ocean, in the soil beneath our feet, or in the air we breathe, cells are cycling endlessly, contributing to the balance of ecosystems. When one contemplates this, it becomes clear that the cell cycle is not just a cellular mechanism, but a principle of life itself. It represents both renewal and continuity, the passing of information from one generation of cells to the next. Without it, living things would remain fixed in time, unable to grow, unable to heal, unable to reproduce. With it, they are dynamic and resilient, able to adapt and persist. There is also a certain elegance in the balance of the cycle. It is not simply about division, but about timing, about knowing when to rest and when to act. In this sense, the cell cycle mirrors life itself, a pattern of activity and repose, of work and renewal. Even at the smallest scale, nature has embedded rhythm and order, ensuring that growth is guided, not chaotic. As we continue deeper into the exploration of cell division, we will look closely at the detailed stages of mitosis and meiosis, observing how the choreography of chromosomes unfolds. But before entering those specifics, it is soothing to remain with the broader view of the cycle as a whole. It is a gentle circle, a continuous path, sustaining all living things through its quiet repetition. It is a reminder that beneath every visible form, from the simplest to the most complex, there exists a patient rhythm, the cell cycle, endlessly shaping and renewing the fabric of life. 3. Interphase. The resting preparation. When people first hear the word interphase, they might imagine a pause or an intermission, as if the cell has stepped away from its duties to rest. Yet this stage, which takes up the vast majority of a cell's life, is anything but idle. Interphase is a time of quiet preparation, a time when the cell grows, gathers resources, and carefully copies its genetic instructions. It is in many ways the most important portion of the cell cycle, because without the long and steady work accomplished during interphase, the dramatic moment of cell division could not succeed. The activities of interphase are subtle, not as easily observed under a microscope as the bold movements of chromosomes during mitosis, but they are absolutely essential to the continuity of life. Imagine a bustling workshop behind closed doors. From the outside, it may appear silent, yet within, machines are humming, materials are being assembled, and intricate plans are being followed with care. Interphase is that hidden workshop. The cell is preparing itself for a moment of duplication, making certain that when the time comes to divide, each daughter cell will receive all it needs to live and function. Interphase is traditionally divided into 3 parts: G1, S, and G2. These names may sound technical, yet their meanings are straightforward once considered slowly. G1, the first gap, is a period of growth. Here, the cell enlarges, taking in nutrients, building proteins, and expanding its internal structures. It is much like a child growing into an adult, accumulating the strength and resources needed for future tasks. During G1, the cell is also highly active in its normal work. A liver cell might process toxins, a muscle cell might contribute to movement, and a skin cell might produce keratin. The cell does not stop performing its role simply because it is preparing to divide. Instead, it balances its ongoing tasks with the quiet accumulation of materials that will be needed for duplication. The S phase, or synthesis phase, is the central event of interphase. In this stage, the DNA is copied. This is no small feat, for DNA is immensely long and intricately organized. Within the nucleus of each cell, strands of DNA form the instructions for every aspect of life. To ensure that both new cells after division will have a complete set of instructions, the DNA must be duplicated with astonishing accuracy. Specialized enzymes act as caretakers in this process. They unwind the double helix, separate the two strands, and guide the assembly of new complementary strands. This process of replication proceeds along the DNA like a slow but steady tide, ensuring that every base, every letter of the genetic script is faithfully recorded. Errors are rare, but the cell has proofreading mechanisms to correct them, like a careful editor reviewing each line of text before moving on. Once DNA has been replicated, the cell enters the G2 phase, the second gap. This is a final period of preparation before the leap into mitosis. The cell continues to grow and begins to produce the proteins and structures needed for chromosome movement. The mitotic spindle, a framework of fibers that will later pull chromosomes apart, requires the components that are assembled during G2. The cell checks its work, verifying that DNA has been copied without significant damage, and ensuring that energy reserves are sufficient. Only once these conditions are satisfied will the cell be permitted to move forward into division. One of the most remarkable aspects of interphase is its balance between continuity and preparation. On the one hand, the cell is living out its specialized role, contributing to the organism as a whole. On the other hand, it is quietly preparing for the future, laying the groundwork for another generation of cells. This duality gives interphase a special character. It is not dramatic like mitosis, where chromosomes are visibly shuffled and separated, but it is filled with steady, unseen labor. It is also worth noting that not all cells proceed through interphase in the same way. Some cells, such as certain nerve cells in the human brain, enter a long-term resting state called G0 after completing G1. In this state, they withdraw from the cycle and may remain without dividing for years or even a lifetime. These cells still carry out their functions. They transmit signals, they process information, but they no longer prepare for division. Other cells, such as those in the lining of the gut or the surface of the skin, move swiftly through interphase, dividing frequently to replenish tissues that are worn away or damaged. In this way, interphase adapts to the needs of the organism, lengthening or shortening depending on context. The importance of interphase becomes even clearer when one considers what happens if the process is disrupted. If DNA is not replicated correctly during the S phase, or if damage is overlooked in G2, the result can be harmful mutations passed on to daughter cells. That is why cells maintain checkpoints throughout interphase. These checkpoints are like guardians standing at the borders of each phase, asking whether the work has been done properly and whether it is safe to proceed. If a problem is detected, the cell may pause to make repairs. If the damage is too severe, the cell may even initiate a process of programmed death, called apoptosis, to prevent errors from spreading. Such decisions may sound dramatic, but they highlight the wisdom of nature's design. It is better for a single cell to sacrifice itself than for errors to multiply throughout the body. Interphase is also deeply connected to the rhythm of life we can observe on a larger scale. Each new leaf, each ring of wood, each root exploring deeper into the soil depends on the multiplication of cells. But before those cells divide, they spend long hours, days, or even weeks in interphase, copying their DNA, building resources, and preparing themselves for the act of duplication. The towering oak or the flowering cherry is possible only because, quietly and faithfully, interphase has done its work within countless cells. In animals, too, interphase sustains growth and healing. When a child scrapes a knee, Skin cells near the wound enter interphase, replicate their DNA, and prepare to divide, gradually filling in the gap until the surface is restored. When a bone is broken, cells in the marrow and surrounding tissues move through interphase, producing new material to knit the pieces together. Even in the absence of injury, interphase sustains the steady turnover of tissues, blood cells renewing every few weeks, hair growing strand by strand, and the lining of the stomach replenishing itself after daily wear. Each of these processes begins not with visible division, but with the quiet preparation of interphase. There is a calming reassurance in knowing that most of a cell's life is spent in this phase of preparation. It reminds us that growth is not always about dramatic leaps forward, but often about the steady, unseen gathering of strength. Just as a forest prepares in winter for the burst of spring, so too does a cell prepare in interphase for the moment of division. And just as the beauty of spring depends on the patience of winter, the success of cell division depends on the careful work of interphase. As we reflect on this stage, we might imagine interphase as the gentle breath before action, the pause before music begins, the quiet laying of foundations before a structure rises. Though it may not draw attention in the same way as mitosis, it carries a weight of importance that cannot be overstated. Interphase is the hidden strength of the cell cycle, the steady hand that ensures order, accuracy, and readiness. Without it, division would be reckless and unstable. With it, division becomes a precise act of renewal, sustaining life through countless generations of cells. And so, while interphase may be called a resting phase, it is in truth a time of constant activity, of careful preparation, and of quiet persistence. It is the patient work behind the scenes, the labor that ensures continuity. Every living thing, from the smallest bacterium to the most complex human body, depends on this long and quiet preparation. Interphase may be unseen, but it is always present, guiding the cycle of life forward with its steady and dependable rhythm. 4. Mitosis overview. When the quiet preparation of interphase comes to a close, the cell is finally ready to take the next great step in its journey, the process of mitosis. This is the stage that most people imagine when they think of cell division, for it is here that the cell's genetic material becomes visible, gathering into neat structures and then parting ways with a graceful choreography. Mitosis is the art of distributing DNA, ensuring that each new daughter cell receives a complete and accurate copy of the genetic instructions that guide life. It is a process that unfolds in stages, each with its own purpose, each building upon the last, and together they form a cycle of continuity that has been carried out across countless generations of cells. Mitosis might be compared to the performance of a quiet but important ritual. The cell begins by carefully condensing its DNA, which had previously been spread out like a soft web within the nucleus. In this condensed form, the DNA takes on the familiar shape of chromosomes, structures that can be moved and separated with precision. It is not that new information has been added or taken away, but simply that the information has been reorganized into a form more suitable for distribution. Much as a librarian might place scattered notes into neat stacks before passing them along, the cell organizes its DNA into tidy packages so that nothing will be lost or misplaced. The stages of mitosis, prophase, metaphase, anaphase, and telophase follow one another. Each stage carries the process forward, guiding chromosomes from a state of unity to a state of separation. Though we give each stage a name, the transitions are gentle, flowing one into the next, without abrupt interruption. Together, they form a single, continuous act of division. In the early moments of mitosis, the nuclear envelope that once shielded the DNA begins to dissolve, opening the way for chromosomes to move freely. Meanwhile, structures known as spindle fibers begin to assemble, stretching across the cell like slender threads. These fibers will serve as guides and anchors, ensuring that chromosomes are pulled to their rightful destinations. It is remarkable to consider that these fibers, though invisible to the naked eye, have the strength and coordination to move entire sets of genetic instructions with accuracy. As the process continues, the chromosomes align themselves in the center of the cell. This stage, called metaphase, is often described as the most striking moment of mitosis, for the chromosomes form a line across the middle, poised and balanced. Each chromosome is attached to spindle fibers from opposite sides of the cell, as if each were being gently held by two hands. This arrangement ensures that when the time comes to separate, each side will receive exactly one copy of every chromosome. It is a simple yet elegant solution to the challenge of distribution. Following this, the fibers begin to shorten, drawing the paired chromosomes apart. This is anaphase, the moment when the two identical sets of DNA begin to move away from one another, slowly, steadily. They are pulled toward opposite ends of the cell, like travelers departing for different destinations. The symmetry is striking. One set of instructions goes to the left, the other to the right, and in this way, the cell ensures equality between its two daughters. As the chromosomes reach their destinations, the cell begins to restore order. A new nuclear envelope forms around each set of DNA, enclosing it once more in a protective boundary. The chromosomes, no longer needing to remain tightly packed, begin to unravel, spreading out again into the thread-like form suited for everyday life. This is telophase, the stage in which the drama subsides, and the two new nuclei stand complete, each holding a full copy of the genetic script. Yet mitosis alone does not complete the process. It must be followed by cytokinesis, the division of the cell's cytoplasm. This is the final act, where the single cell physically separates into two. A contractile ring, made of filaments, tightens around the middle of the cell, pinching it until it cleaves in two. When the process is complete, what was once one cell has become two, each with its own nucleus, its own cytoplasm, and its own share of the original cell's organelles. These two daughter cells are, in most cases, identical to one another and to the parent cell, ready to begin the cycle anew. The elegance of mitosis lies not only in its precision but also in its universality. From the skin cells on your hand to the cells that form the lining of a leaf, mitosis occurs in much the same way. It is a pattern, repeated across life, a fundamental solution to the challenge of continuity. Wherever multicellular organisms grow, heal, or maintain themselves, mitosis is quietly at work. Consider how often this process occurs within your own body. Trillions of cells make up the human form, and every day, billions of them divide. Without mitosis, the skin could not renew itself, blood could not be replenished, and wounds could not be healed. It is through this silent choreography that life maintains itself, moment after moment, year after year. And yet, as perfect as mitosis may seem, it is not a reckless rush forward. At every stage, safeguards exist to ensure that the process is proceeding correctly. If chromosomes do not align properly during metaphase, the cell will pause, delaying the separation until the issue is resolved. If DNA has been damaged before division, the cell may halt mitosis entirely, returning to repair mechanisms or choosing not to divide at all. This careful oversight prevents errors, for even small mistakes in the distribution of chromosomes can have significant consequences. There is something deeply calming in the thought that mitosis is not hurried but measured. It is not an accident of nature but a carefully orchestrated cycle, honed by billions of years of evolution. Each step flows into the next. Each structure knows its place, and the result is the faithful passing on of life's instructions. When we pause to reflect on mitosis, we see more than just the movements of chromosomes. We see the very principle of renewal, the quiet assurance that life will continue. Each time a cell divides, it is as though a promise has been kept, a promise that the story of life will not end with one cell, but will go on, branching into two, and then into many more. It is a cycle of continuation, and in its simplicity lies its beauty. As we continue our journey through the cell cycle, it is helpful to hold this image of mitosis in mind, a steady choreography, a quiet dance of chromosomes, an act of balance and renewal. In the next reflections, we will look more closely at each of its stages, seeing how the details unfold. But for now, it is enough to rest with the overview, the gentle knowledge that mitosis is the mechanism by which life ensures its own continuity, softly and endlessly repeating, sustaining the delicate fabric of existence. 5. Prophase, setting the stage. When a cell has completed its long and patient preparation in interphase, and when the cycle gently carries it forward into the act of division, the first stage that greets it is called prophase. This is the moment when the quiet interior of the cell begins to shift, when structures that once seemed calm and ordinary transform into the ordered patterns required for the careful sharing of genetic material. Prophase is often considered the beginning of mitosis proper, and though it is just one part of the larger journey, it holds a special significance because it sets the stage for everything that follows. Without the careful events of prophase, the separation of chromosomes later in the cycle could not succeed. If you imagine the nucleus of a cell during interphase, the DNA is spread out in a delicate and almost invisible state, like threads of mist drifting within the nuclear space. In this form, the genetic material is accessible for reading and copying, but it is not well suited for movement. Prophase begins when those threads begin to coil and condense, gathering themselves into compact chromosomes. This condensation is not random but highly organized, with each chromosome becoming a clearly defined structure, strong enough to be moved and divided. Under a microscope, this is the first moment when chromosomes appear as distinct entities rather than diffuse clouds. It is as though the cell is gathering its thoughts, preparing its most precious instructions for the journey ahead. Each chromosome, it should be remembered, has already been duplicated during the synthesis stage of interphase. Thus, in prophase, each chromosome is actually made of two identical sister chromatids, joined at a narrow region called the centromere. These chromatids are like twin scrolls of information, perfectly copied and waiting to be distributed to future daughter cells. The centromere holds them together for now, but soon it will serve as the attachment site for the fibers that will guide them apart. While the chromosomes condense, other changes are also beginning. The nucleolus, that small and often overlooked structure within the nucleus where ribosomes are assembled, gradually fades from view. Its disappearance signals that the cell is shifting priorities, turning away from ordinary protein production and devoting itself fully to the act of division. At the same time, the nuclear envelope, the protective membrane that surrounds the nucleus, begins to break down. This dissolution is a careful process, opening the way for chromosomes to interact with structures outside the nucleus. Without this change, the chromosomes would remain confined and unable to move freely. Beyond the nucleus, the cell is busy preparing the machinery required for chromosome movement. Structures known as centrosomes, each containing a pair of centrioles, begin to migrate toward opposite poles of the cell. From these centrosomes, microtubules extend outward, forming the beginnings of what will become the mitotic spindle. The spindle is an intricate framework of fibers that will later attach to chromosomes and guide them with precision. In prophase, it is only beginning to take shape, stretching like fine lines of scaffolding across the cell. Though fragile in appearance, this structure is essential for the proper alignment and eventual separation of chromosomes. It is worth pausing here to appreciate the subtle coordination of these events. Prophase is not simply about condensing DNA or building spindles in isolation, but about weaving these changes together into a coherent whole. The condensation of chromosomes ensures they can be moved. The breakdown of the nuclear envelope ensures they can reach the spindle, and the migration of centrosomes ensures that the cell will have two opposing poles toward which chromosomes can be guided. Each step anticipates the next, like careful brushstrokes in the first stage of a painting that will later come alive with detail. Under the microscope, the cell in prophase begins to look quite different from one in interphase. The once transparent nucleus now contains visible chromosomes, each appearing as an X-shaped structure of two sister chromatids. The nuclear envelope grows faint, and the spindle fibers stretch like delicate lines across the cell's interior. To the observer, it is as though the cell has shifted from a quiet resting state into a poised readiness, every structure preparing for the choreography to come. Prophase also gives way to a brief transitional stage, often called prometaphase, where the breakdown of the nuclear envelope is complete and spindle fibers fully attached to chromosomes at specialized sites called kinetochores. These kinetochores are tiny protein structures located at the centromeres of chromosomes, acting like handles that spindle fibers can grasp. Once attached, the fibers can begin to exert gentle tension, testing their connections, as if ensuring that every chromosome is properly linked before the grand alignment of metaphase. Though prometaphase is sometimes distinguished as a separate stage, it is in truth a natural continuation of prophase, another step in setting the stage for accurate division. The importance of prophase cannot be overstated. If chromosomes failed to condense, they would tangle and tear during movement. If the nuclear envelope failed to break down, the chromosomes would remain trapped, unable to align or separate. If spindle fibers failed to form or attach properly, chromosomes could be lost or distributed unevenly. These errors would carry consequences, potentially leaving daughter cells with incomplete or incorrect sets of instructions. Prophase prevents these problems by ensuring that everything is in place before the critical act of separation begins. One might find comfort in the thought that nature has invested so much care in this stage. It is not rushed, not careless, but deliberate and precise. The transformations of prophase are protective measures, safeguarding the integrity of genetic information. Each change serves a purpose. Each adjustment ensures balance. In the quiet orderliness of this stage, there is a lesson about preparation itself, that before any great action, careful groundwork must be laid. The universality of prophase is another source of wonder. Whether in the cells of a plant root tip or in the dividing cells of animal tissue, the same sequence unfolds. Chromosomes condense, the nuclear envelope breaks down, the spindle forms, and the cell prepares for the equal sharing of its instructions. Across all of life, in countless organisms and environments, prophase quietly carries out its task, unseen yet indispensable. And beyond the biology, there is also a poetic sense in which prophase represents anticipation. It is a beginning, but not yet the climax. The chromosomes are gathered, but not yet moved. The spindle is built, but not yet tested. The scene is prepared, but the performance is still to come. It is the moment before action, filled with potential, charged with quiet energy. In this way, prophase mirrors the rhythms of life itself, reminding us that much of existence is preparation, the slow and steady laying of foundations upon which moments of change can unfold. When we reflect on prophase in this way, it becomes more than just a stage of mitosis. It becomes a reminder of the importance of readiness, of the value of preparation, of the beauty that can be found in the subtle shifts that make great changes possible. It is not the most dramatic stage, but it is one of the most essential, without which the entire choreography of division would falter. As the cell completes prophase, it stands ready for what comes next. Chromosomes are condensed and visible, spindle fibers are stretching across the cell, centrosomes are positioned at opposite poles, and the nuclear envelope has dissolved to make way for movement. The stage is fully set, and the cell waits to carry its chromosomes into alignment at the center. Soon, metaphase will follow, and the symmetry of division will be revealed. But before that, it is worth pausing in appreciation of this stage, the quiet yet crucial work of prophase, in which the cell prepares itself for the delicate and enduring act of renewal. 6. Metaphase and anaphase. Once the careful preparations of prophase are complete, the cell moves onward into a stage of extraordinary symmetry and balance, followed by a moment of graceful separation. These two stages, metaphase and anaphase, are often spoken of together, for they flow seamlessly from one into the other, each representing a turning point in the process of mitosis. In metaphase, the chromosomes align with quiet precision at the center of the cell, forming a line that is both orderly and striking. In anaphase, that symmetry is gently undone as the identical halves of each chromosome are drawn apart, carried to opposite sides of the cell. Together, these stages embody the twin principles of balance and division, ensuring that life's instructions are passed on faithfully to each daughter cell. To understand metaphase, imagine the cell as a stage, and the chromosomes as actors who must take their positions before the performance can proceed. During prophase and prometaphase, the chromosomes condensed and attached to spindle fibers. Now, in metaphase, those fibers draw each chromosome into alignment along the metaphase plate, an invisible plane that runs through the center of the cell. This arrangement is not random. It is a carefully orchestrated alignment that ensures every chromosome is positioned correctly. Under a microscope, the sight is striking. The chromosomes form a neat line across the middle of the cell, each one poised like a figure awaiting its cue. This moment of alignment is not just for appearances, it serves a critical purpose. By lining up along the metaphase plate, chromosomes ensure that when they are finally separated, each daughter cell will receive exactly one copy of every chromosome. The spindle fibers that hold them are attached to kinetochores, protein structures located at the centromeres of each chromosome. From opposite poles of the cell, fibers pull gently, creating tension on the chromosomes. This tension is carefully monitored by the cell, and it serves as a signal that everything is ready. Only when all chromosomes are properly aligned and under balanced tension does the cell allow itself to move forward. In this way, metaphase acts as a checkpoint, a final moment of reassurance that the division will unfold correctly. There is something calming in the thought of metaphase as a pause for balance. The chromosomes, having traveled to the center, wait in equilibrium, neither moving forward nor slipping back. It is as if the cell itself is taking a breath, making certain that all is in harmony before the great act of separation begins. This stage can last longer than one might expect, as the cell does not rush. It waits until every chromosome is properly aligned, until the spindle fibers are perfectly secured, until the conditions are just right. Only then does it proceed, confident that the division will be accurate and complete. When the moment arrives to move forward, the transition into anaphase begins. The centromeres, which until now have held the sister chromatids together, split apart. Each chromatid, once joined with its twin, is now free to travel independently. What were once pairs of chromatids are now individual chromosomes, each destined for a different daughter cell. The spindle fibers shorten, pulling these chromosomes toward opposite poles of the cell. It is a steady, deliberate movement, not hurried, but sure. Under the microscope, one can see the chromosomes being drawn away from the center, spreading apart in two symmetrical groups. Anaphase is the moment of true division, the point at which the genetic material is physically separated. The precision of this process is remarkable. Every chromosome that was lined up in metaphase now finds its way to one side or the other, guided by the spindle fibers. The forces at work are microscopic, yet they carry with them the weight of life's continuity. If even one chromosome were left behind, or if one side received an extra, the balance would be broken, and the resulting cells might suffer. Yet in most cases, the process is flawless. a testament to the care with which evolution has shaped it. As the chromosomes move apart, the cell itself begins to elongate, stretched by the spindle fibers that push against one another. This elongation helps to separate the two groups of chromosomes even further, creating the space that will later allow two distinct nuclei to form. The image of anaphase, with chromosomes streaming toward opposite poles, is one of the most iconic in all of cell biology. It is the very embodiment of division, the moment when one becomes two, not yet fully separated. It is soothing to think of metaphase and anaphase as a balance of opposites. In metaphase, the chromosomes are gathered together, united in a single line. In anaphase, they are separated, moving away from one another in mirrored groups. One stage emphasizes harmony, the other emphasizes division. Yet both are necessary, and both are guided by the same underlying purpose: to ensure that each new cell receives a complete and faithful copy of the genetic instructions. Without the alignment of metaphase, the separation of anaphase would be unreliable. Without the separation of anaphase, the balance of metaphase would serve no purpose. Together, they complete the central act of mitosis. Beyond the microscopic details, these stages also carry a deeper resonance. They remind us that life depends on both unity and separation, on moments of gathering and moments of parting. Just as families come together and then branch apart, or as tides flow inward and outward, so too do chromosomes align and then separate. The cell cycle mirrors the rhythms of nature, reflecting the balance between holding together and letting go. In this way, metaphase and anaphase are not just stages of biology, but symbols of the universal patterns that shape existence. As anaphase comes to a close, the chromosomes draw near to the poles of the cell. Each side now holds a complete set, identical to the other, and the groundwork has been laid for the final steps of mitosis. Soon, telophase will follow, bringing restoration and renewal, as new nuclei form around the chromosomes and the cell prepares to divide into two. But before moving ahead, it is worth pausing with the image of metaphase and anaphase in mind. The perfect line of chromosomes in balance, followed by the graceful movement apart. These stages embody the quiet precision of life at its smallest scale, a reminder of the hidden order that sustains the living world. In the silence of a single cell, without sound or spectacle, metaphase and anaphase unfold countless times each day, not only in humans but in every living organism that grows, heals, or renews itself. The forest tree, the flowing river algae, the tiniest insect, all are sustained by these hidden movements. To know this is to glimpse the continuity of life itself, carried out in the smallest of spaces, yet with consequences that span the vastness of generations. 7. Telophase and cytokinesis. When the elegant separation of anaphase has run its course, and the identical sets of chromosomes have been guided to opposite ends of the cell, a new stage begins. One that is calmer in its character but no less important. This stage is telophase, followed closely by cytokinesis, and together they mark the quiet conclusion of mitosis. If prophase was the preparation, metaphase the alignment, and anaphase the decisive separation, then telophase and cytokinesis are the restoration, the gentle closing of the cycle, and the beginning of life anew for the daughter cells that emerge. In telophase, the changes that carried the cell into division begin to reverse. The condensed chromosomes, which during prophase had coiled into tight and visible shapes, now begin to uncoil, returning to their more relaxed and thread-like form. In this unwound state, they will once again be accessible for the reading and copying of genetic information during interphase. To an observer at the microscope, the distinct X-shaped structures of the chromosomes gradually fade into a softer, more diffuse appearance, signaling that the intensity of mitosis is coming to a close. At the same time, The nuclear envelope begins to reform around each set of chromosomes. During prophase, this protective boundary had dissolved to allow the chromosomes freedom of movement. Now, during telophase, new membranes assemble around each group, creating two separate nuclei within the cell. This act of enclosure is symbolic of a return to order. The genetic instructions are once again safely housed, each nucleus containing a full and identical copy. Inside these newly formed nuclei, the nucleolus reappears, a small but vital structure where ribosomes will soon be assembled once more. The cell, in a sense, is returning to its everyday life, even as the final division of the cytoplasm remains to be completed. Meanwhile, the spindle fibers that had guided the chromosomes now begin to disassemble. Their task is finished, and their presence is no longer required. The centrosomes and microtubules that formed the spindle return to a more ordinary state, leaving behind the two sets of chromosomes, now enclosed within their new nuclei. The cell is no longer pulled in two directions, but instead is quietly preparing to become two separate entities. This preparation culminates in cytokinesis, the process by which the cytoplasm itself divides, creating two distinct daughter cells. In animal cells, this begins with the formation of a contractile ring composed of actin filaments. Just beneath the cell membrane, the ring tightens like a drawstring, pulling the membrane inward until it pinches the cell into two. This inward pinching is called a cleavage furrow, and as it deepens, the connection between the two halves narrows until finally it separates completely. The result is two cells, each surrounded by its own complete membrane, each with its own nucleus, and each ready to embark upon interphase. In plant cells, cytokinesis takes a slightly different form. Because plant cells possess rigid cell walls, they cannot be pinched apart in the same way as animal cells. Instead, small vesicles, filled with cell wall materials, gather at the center of the cell. These vesicles fuse together to form what is called the cell plate, which expands outward until it reaches the cell's edges, dividing the cytoplasm and creating a new cell wall between the daughter cells. Whether in animals or plants, the result of cytokinesis is the same. Two distinct daughter cells, each a mirror of the other, each containing the full instructions for life. It is soothing to reflect on how telophase and cytokinesis bring closure to the dramatic events of mitosis. Earlier stages were filled with movement and tension, the condensation of chromosomes, their alignment along the metaphase plate, and their pulling apart during anaphase. Telophase, by contrast, is a stage of restoration. It is as if the cell, having completed a demanding task, now takes a deep breath, uncoiling its DNA, rebuilding its nuclei, and returning to a state of calm. Cytokinesis follows as the final gesture, a graceful separation that leaves behind two new cells where once there was only one. This conclusion is not the end of the story, but a renewal. Each daughter cell will now enter interphase, where it will grow, perform its specialized tasks, and prepare in time for its own cycle of division. The continuity of life is preserved not through grand and sudden changes, but through this repeating cycle of division and renewal, carried out steadily within every living organism. The significance of telophase and cytokinesis becomes even clearer when one considers the balance they provide. Without telophase, the chromosomes would remain condensed, inaccessible for the work of transcription and replication. Without cytokinesis, the division of genetic material would remain incomplete, leaving behind a cell with two nuclei, but no true separation. It is only by completing both stages that mitosis achieves its purpose, the creation of two fully independent daughter cells, each ready to continue the rhythm of life. What is perhaps most striking about telophase is its sense of symmetry. At the start of mitosis, there was one nucleus, housing a complete set of genetic instructions. By the end of telophase, there are two nuclei, each identical to the original, each containing the same information. The fidelity of this process is astonishing, for within those nuclei are thousands of genes, encoded along long strands of DNA, and yet they are passed on with near-perfect accuracy. This precision is not accidental, but the result of countless safeguards built into the cell cycle. It is a quiet testament to nature's care that such a delicate process is carried out with such reliability. Cytokinesis, too, carries a sense of balance and beauty. It is the moment when one cell truly becomes two, not just in terms of nuclei, but in terms of physical separation. The actin ring in animal cells, tightening with gentle force, and the cell plate in plant cells, forming patiently from vesicles, both embody the same principle. That division must be complete for renewal to be possible. Each daughter cell must be independent, with its own space, its own cytoplasm, and its own ability to grow and thrive. There is comfort in knowing that this process unfolds countless times each day within our own bodies. Each second, millions of cells complete telophase and cytokinesis, renewing tissues, repairing damage, and sustaining life. The skin that heals after a cut, the blood cells that replenish after loss, the lining of the stomach that restores itself, all depend on these final stages of mitosis. Without them, renewal would remain unfinished, and continuity would be broken. Beyond the biology, there is also something symbolic in the quiet resolution of telophase and cytokinesis. They remind us that every cycle of effort must come to a close, that every act of division must find balance in restoration. Just as a day concludes with evening and the passing of light into rest, so too does mitosis conclude with telophase, where the intensity of division gives way to calm. And just as night gently yields to the dawn of a new day, so too does cytokinesis give rise to two new cells, each beginning again in interphase, each ready to carry forward the rhythm of life. In the great continuity of existence, telophase and cytokinesis represent closure and renewal, endings that are also beginnings. They are the quiet assurance that life will not end with one cell, but will continue with two, and then with many more, endlessly repeating across generations and across time. To picture these stages is to glimpse the elegance of life at its smallest scale, a hidden symmetry unfolding in silence, yet carrying within it the persistence of all living things. 8. Meiosis, division for reproduction. When we think of cell division, mitosis often comes first to mind, for it is the cycle that renews our tissues, repairs wounds, and maintains life on a daily basis. Yet, there exists another form of division, one that is equally essential, but directed toward a very different purpose. This process is meiosis, and it is the quiet architect of reproduction in sexually reproducing organisms. If mitosis sustains the body, meiosis ensures the continuation of the species, creating cells that can unite in the act of fertilization to form new life. Its rhythm is more complex than that of mitosis, involving 2 consecutive rounds of division, yet its outcome is elegant. Gametes, such as sperm or egg cells, each containing half the genetic material of the parent. Through this halving and subsequent recombination, meiosis introduces both continuity and variation, 2 forces that together shape the diversity of life. To understand why meiosis is necessary, it is helpful to reflect on the nature of chromosomes. In most organisms, chromosomes come in pairs, one set inherited from the mother, the other from the father. In humans, this means 46 chromosomes, arranged as 23 pairs. If gametes were produced by simple mitosis, they too would contain 46 chromosomes. When 2 such gametes fused at fertilization, the resulting zygote would contain 92 chromosomes, and the number would double again with each generation. Clearly, such a system could not be sustained. Instead, meiosis solves this problem by halving the chromosome number. Gametes carry only 1 chromosome from each pair, so when they unite, the zygote is restored to the proper total. This balance ensures that the chromosome number remains constant across generations, neither doubling nor diminishing with time. The process of meiosis is both similar to and distinct from mitosis. Like mitosis, it begins with the replication of DNA, so that each chromosome consists of two identical sister chromatids. But unlike mitosis, meiosis involves two successive divisions, known as meiosis I and meiosis II, each with its own stages. The result of these two rounds is 4 daughter cells, each with half the number of chromosomes. This reduction is the key to sexual reproduction, making possible the fusion of gametes without disrupting genetic balance. What makes meiosis particularly fascinating is not only the halving of chromosomes but also the opportunities it creates for variation. During the early stages of meiosis I, homologous chromosomes pairs, consisting of one maternal and one paternal chromosome, come together in a process called synapsis. They align side by side, forming structures known as tetrads. Within these tetrads, segments of DNA may be exchanged in an event called crossing over. This exchange shuffles genetic material, creating new combinations of genes that were not present before. Later, when the chromosomes are separated, the assortment of maternal and paternal chromosomes is essentially random. These mechanisms, crossing over and independent assortment, introduce diversity into gametes, ensuring that no two are exactly alike. It is this diversity that underlies the vast variety we see in living organisms, from the patterns of leaves on trees to the traits of siblings within the same family. To follow meiosis more closely is to witness a more intricate dance than mitosis. In meiosis, homologous chromosomes pair, exchange segments, and then separate into two cells, each still containing duplicated chromatids, but only half the number of chromosome pairs. In meiosis II, these chromatids are finally pulled apart, much like in mitosis, producing 4 cells in total. Each of these cells is haploid, carrying only one set of chromosomes, ready to participate in fertilization. Though the mechanics can appear complex, the underlying principle is simple: to create gametes that preserve genetic balance and allow for variation. Consider the significance of this. Without meiosis, sexual reproduction would not be possible in its present form. Gametes would not be reduced to half, and the union of cells would quickly disrupt the stability of chromosome numbers. More importantly, without the variation introduced by meiosis, populations would lack the diversity that makes adaptation possible. Evolution, in its gradual unfolding, depends upon this diversity, for it provides the raw material upon which natural selection acts. Meiosis, then, is not just a mechanism of reproduction but also a source of the endless variety of life. In humans and other animals, meiosis occurs in specialized tissues. In males, it takes place in the testes, producing sperm cells. In females, it occurs in the ovaries, producing egg cells. In plants, meiosis gives rise to spores, which then develop into gametophytes that ultimately produce gametes. Across all these forms, the outcome is the same: cells that carry half the genetic information, ready to unite and form something new. One might pause here to reflect on the beauty of meiosis as a balance of continuity and change. On the one hand, it ensures continuity, passing on the essential genetic instructions from parent to offspring. On the other hand, it introduces change, mixing and reshuffling these instructions so that each new individual is unique. This duality, preserving what must endure while allowing variation, lies at the heart of life's resilience. It explains why siblings, though similar, are never identical, and why populations can adapt to shifting environments. The quiet work of meiosis often goes unnoticed, yet its impact is everywhere. Each person, each animal, each plant that reproduces sexually owes its existence to the orderly unfolding of meiosis in the generation before. It is humbling to think that within the ovary or the testis, within the flower or the spore, countless cells are carrying out this intricate sequence, producing gametes that hold the potential for entirely new beings. Every seed, every newborn, every hatchling is a testament to the careful rhythm of meiosis. And just as mitosis carries lessons about renewal and balance, meiosis carries lessons about diversity and unity. It shows us that new life emerges not by simple copying but by combining and reshuffling, by bringing together contributions from two parents to create something new. In this way, meiosis reflects the principle that strength often lies in combination, that richness arises from diversity, and that continuity is not the repetition of sameness, but the weaving together of differences. As meiosis concludes, the 4 haploid cells it produces stand ready for their role. They may remain dormant for a time, or they may proceed quickly toward fertilization, depending on the organism. But whatever their fate, their existence ensures the persistence of life through generations. They are the seeds of continuity, carrying within them both the legacy of the past and the potential of the future. Thus, while mitosis and meiosis may appear similar at first glance, their purposes set them apart. Mitosis is the cycle of maintenance and repair, the quiet engine of daily life. Meiosis, by contrast, is the cycle of renewal across generations, the bridge between parents and offspring, the source of diversity in the living world. Together, they form the twin foundations of cellular reproduction, one sustaining the present, the other ensuring the future. In the silence of cells, without announcement or spectacle, meiosis unfolds again and again, producing the gametes that carry life forward. To contemplate it is to see not just the mechanics of chromosomes, but the deeper principle of continuity through change. It is a gentle reminder that every being, no matter how unique, is the product of this hidden cycle, and that the variety of life around us is rooted in the quiet movements of chromosomes during meiosis. 9. The importance of checkpoints. The cell cycle, with its steady rhythm of preparation, division, and renewal, may seem almost mechanical in its repetition, yet beneath its regularity lies a system of remarkable vigilance, a quiet series of safeguards known as checkpoints. These checkpoints act as guardians of the cycle, pausing the process at crucial moments to ensure that conditions are right, that DNA is intact, and that each step has been carried out with care. In this way, the cell avoids errors that might otherwise threaten its survival, or the survival of the organism as a whole. If mitosis and meiosis are the performances, then checkpoints are the stage managers, watching every detail and allowing the process to continue only when everything is in order. To understand the importance of checkpoints, one might imagine a journey along a winding path. At intervals, there are gates where the traveler must pause. At each gate, a watchful figure asks whether the traveler is prepared, whether they carry what they need, whether the road ahead is safe. Only when the answers are satisfactory does the gate open. In much the same way, checkpoints in the cell cycle allow the cell to proceed only when it is ready. If there is a problem, the gate remains closed until it is resolved, or until the cell chooses another path. There are 3 main checkpoints in the cycle, each positioned at a critical moment. The first lies in the G1 phase, before the cell commits to DNA replication. The second lies at the end of G2, before the cell enters mitosis. The third lies during metaphase, when chromosomes must be correctly aligned before they can be separated. Each of these checkpoints asks a different set of questions, yet all share the same purpose: to protect the integrity of the genetic material and to ensure that division will produce healthy daughter cells. At the G1 checkpoint, sometimes called the restriction point, the cell pauses to consider its environment and its own readiness. Has it grown large enough? Does it have sufficient nutrients and energy to proceed? Is the DNA free of damage? If the answers are yes, the cell moves forward into the S phase, where DNA will be replicated. If not, the cell may delay, repairing damage or gathering resources before trying again. In some cases, if conditions are unfavorable for too long, the cell may exit the cycle entirely and enter a resting state called G0. There it can remain quietly for days, years, or even a lifetime, carrying out its functions without dividing. This checkpoint is therefore a point of decision, where the cell weighs its options and chooses a path. The G2 checkpoint comes later, after DNA has been replicated. Here, the cell asks a new set of questions. Has all the DNA been copied completely? Are there any errors or breaks that must be repaired? Are the proteins needed for mitosis in place? If the answers are satisfactory, the cell proceeds into prophase. If not, the cycle pauses, giving time for repair enzymes to correct mistakes. Without this checkpoint, cells might enter mitosis with damaged or incomplete DNA, a dangerous situation that could lead to defective daughter cells. By pausing at G2, the cell ensures that it does not carry errors forward into the next generation. The final major checkpoint occurs during metaphase, at the very heart of mitosis. At this stage, the chromosomes are aligned along the metaphase plate, and spindle fibers are attached to their kinetochores. The checkpoint here, often called the spindle assembly checkpoint, ensures that every chromosome is properly connected to the spindle before separation begins. If even one chromosome is unattached or misaligned, the cell waits. Only when every chromosome is under tension, pulled equally from both sides, does the cell allow anaphase to begin. This vigilance prevents the unequal distribution of chromosomes, a condition known as aneuploidy, which can be harmful or even lethal to cells and organisms. It is calming to consider how patient these checkpoints are. They do not rush the cell forward, but wait until the conditions are right. They act not with urgency, but with steadiness, allowing time for correction and repair. In this way, they mirror the rhythms of nature itself, where growth is often slow and deliberate, where balance is maintained by pauses as well as by movement. Checkpoints also carry within them the possibility of choice. If damage is too severe to repair, the cell may initiate apoptosis, a form of programmed death. This decision, though it sounds severe, is in fact an act of preservation, protecting the organism from the spread of damaged cells. Apoptosis is a quiet and orderly process, not a violent one, and it allows the body to remove cells that might otherwise cause harm. In this way, checkpoints not only guard against error, but also provide an alternative path when correction is impossible. Beyond their immediate function, checkpoints also highlight a deeper truth about life: that continuity requires vigilance. The copying and distribution of DNA are delicate tasks, and without oversight, errors would accumulate quickly. The presence of checkpoints ensures that life's instructions are passed on with remarkable fidelity, generation after generation. The fact that humans and other organisms can live for decades, sustained by trillions of cell divisions, is a testament to the quiet effectiveness of these safeguards. It is humbling to imagine the countless checkpoints occurring in your body at this very moment. In the bone marrow, where blood cells are produced, checkpoints pause to verify that DNA is intact before new cells are released. In the skin, where cells divide to replace those lost each day, checkpoints guard against mistakes. Even in the lining of the stomach, where cells are renewed rapidly, these watchful pauses ensure that renewal does not come at the cost of accuracy. Without these checkpoints, the harmony of the body would quickly be disrupted. There is also a poetic lesson in the presence of checkpoints. They remind us that pauses are not failures, but necessary parts of any cycle. Just as the seasons pause in winter before the burst of spring, or as a traveler pauses at a crossroads before continuing, so too do cells pause to ensure that the way forward is safe. These pauses are not wasted time, but moments of preparation and assurance. They are the reason the cycle can continue without collapse. As we reflect on the importance of checkpoints, it becomes clear that they are as vital as the divisions themselves. They are the quiet guardians of the cell cycle, unseen but essential, ensuring that each stage unfolds with accuracy and care. Without them, the beauty of mitosis and meiosis would falter, undermined by errors. With them, the cycle proceeds smoothly, generation after generation, sustaining the continuity of life. Soon we will turn to the details of DNA replication and repair, the mechanisms that allow cells to copy their instructions and correct their mistakes. But before moving forward, it is worth resting with the thought of checkpoints, those silent pauses, those gentle guardians, those unseen gates along the path of the cell cycle. They embody the principle that progress is strongest when guided by vigilance, and that renewal is safest when tempered with care. 10. Replication in detail. At the heart of every act of cell division lies an extraordinary process, one that allows life's most essential instructions to be faithfully preserved and passed on: the replication of DNA. Before a cell can divide, it must first make a complete copy of its genetic material, ensuring that each daughter cell will inherit the full set of instructions required for survival and function. This replication is not only a practical necessity but also a profound event, for it is the quiet mechanism by which continuity is maintained across generations. Every organism alive today, from the simplest bacterium to the most complex human being, carries within it DNA that has been faithfully copied again and again over billions of years. To dwell on this process is to glimpse one of the most enduring rhythms of life, a hidden cycle of duplication that underlies every act of renewal. DNA itself is a molecule of remarkable beauty and order. It is composed of two strands wound around one another in the shape of a double helix, like a spiral staircase, stretching endlessly into the distance. Along the strands are bases adenine, thymine, cytosine, and guanine arranged in precise sequences that form the code of life. Each base pairs with a specific partner, adenine with thymine, cytosine with guanine. These pairings are the key to replication, for they provide a natural template by which each strand can be copied. If one strand reads adenine, the new strand will place thymine. If it reads cytosine, guanine will follow. In this way, the sequence is reproduced with fidelity, letter by letter, word by word. Replication begins at special sites along the DNA called origins of replication. At these points, proteins attach and gently unwind the double helix, creating small bubbles where the two strands are separated. These bubbles expand outward, forming what are known as replication forks, Y-shaped structures where the strands are actively being copied. As the forks move along the DNA, the entire molecule is gradually opened and duplicated until the entire chromosome has been replicated. At the heart of this process is an enzyme called DNA polymerase, the craftsman of replication. DNA polymerase moves along the separated strands, adding new bases to form complementary strands. It works with remarkable speed and accuracy, placing each base according to the rules of pairing. Yet, DNA polymerase does not act alone. A suite of other enzymes and proteins assist in the process, each contributing its own specialized role. Helicase unwinds the double helix, breaking the bonds between bases. Primase lays down short RNA primers to provide starting points. Ligase seals gaps in the newly formed strands, ensuring that the finished product is continuous and unbroken. Together, these enzymes form a cooperative team, working in harmony to copy life's instructions. Replication is not uniform along both strands, because the two strands of DNA run in opposite directions. One strand can be copied smoothly and continuously, while the other must be copied in fragments. The continuous strand is called the leading strand, and it is built steadily as the replication fork advances. The discontinuous strand, called the lagging strand, is assembled in short segments known as Okazaki fragments. These fragments are later joined together by ligase, forming a seamless whole. Though the process may appear uneven, both strands are ultimately copied with the same precision, ensuring that no part of the genetic code is lost. The fidelity of DNA replication is astonishing. Errors occur only once in every billion bases, thanks to the proofreading ability of DNA polymerase. As it adds new bases, the enzyme checks its work, removing mismatched bases and replacing them with the correct ones. This proofreading is a safeguard against mutation, a way of ensuring that the message of life remains intact. Even so, occasional errors do slip through, and it is these rare mutations that provide the raw material for evolution. In this sense, replication is both conservative and creative, preserving continuity while allowing for the possibility of change. It is humbling to think about the scale of this process. In a human cell, the DNA stretches to nearly 2 meters in length if fully uncoiled. Yet it is contained within a nucleus only micrometers wide. During replication, this vast length must be copied in its entirety, with accuracy maintained across billions of bases. And yet the process is carried out quietly, efficiently, and without our awareness, thousands of times each second in the cells of our bodies. Replication is not only remarkable for its mechanics, but also for its timing. It occurs during the S phase of interphase, a period of preparation before mitosis or meiosis. The cell devotes enormous energy and resources to this task, for without it, division would be meaningless. To divide without replication would be to leave one daughter cell without instructions, an outcome that would bring the cycle to an end. By ensuring that each division is preceded by replication, the cell preserves the continuity of life. Beyond its biological necessity, DNA replication carries a symbolic resonance. It is the act of preservation, the safeguarding of memory across time. Just as a scribe carefully copies a manuscript to ensure that its words endure, so too does the cell copy its DNA, ensuring that the instructions of life are not lost. Every trait, every characteristic, every potential is preserved in this quiet act of duplication. The color of a leaf, the pattern of a bird's song, the shape of a human face, all are passed on because DNA replication has carried the code across generations. There is also a lesson in the cooperation of replication. No single enzyme could accomplish the task alone. It requires helicase to open the helix, primase to mark the starting points, polymerase to build the new strands, and ligase to complete the process. Each plays its part, none dominating the others, all contributing to the whole. This cooperation mirrors the nature of life itself, where complex outcomes arise, not from single forces, but from the harmony of many working together. As we reflect on DNA replication, it is easy to feel a sense of awe at its quiet precision. It is an event that unfolds invisibly within every cell, unseen and unnoticed, yet without it life would come to a halt. Each of us carries within our bodies the product of countless replications, stretching back through the generations to the earliest forms of life on Earth. The continuity of existence, the persistence of species, the unfolding of evolution, all rest upon this hidden cycle of copying. In the stillness of the cell, without noise or spectacle, DNA replication unfolds. Bases are paired, strands are extended, fragments are joined. The double helix, once separated, is restored, and the message of life is preserved. To contemplate this process is to see not just the mechanics of molecules, but the persistence of life itself, carried forward through time by the simple yet profound act of copying. It is one of nature's quiet miracles, a rhythm of duplication that sustains the fabric of existence. 11. DNA repair mechanisms. DNA, though astonishing in its complexity and resilience, is not invulnerable. Each strand of this delicate molecule carries the instructions for life. Yet every day it is subject to countless threats. Errors may occur during replication. Bases may be altered by chemicals. Strands may be broken by radiation. And entire sections may become mismatched or distorted. Left uncorrected, such damage could lead to mutations, disrupting the instructions that guide the cell. But life has endured for billions of years, and one of the reasons it has been able to do so is that cells possess an extraordinary capacity for repair. DNA repair mechanisms work silently and continuously, like watchful caretakers, patrolling the genetic material and correcting mistakes. This constant vigilance preserves the fidelity of inheritance, ensuring that the instructions passed from cell to cell remain accurate and usable. To appreciate the importance of repair, it helps to think about scale. In a single human cell, billions of base pairs of DNA are replicated and maintained. Each base must be correct, each bond intact, for the instructions to make sense. Even if only a small fraction of these bases were altered or lost, the consequences could be severe. Yet cells manage this challenge with remarkable success, correcting the vast majority of errors before they can cause harm. The result is a system that is both flexible and durable, able to withstand damage while maintaining the integrity of life's code. One of the simplest and most frequent forms of repair is proofreading during DNA replication. As DNA polymerase adds new bases to the growing strand, it checks each one against the template. If the wrong base is inserted, the polymerase senses the mismatch, removes it, and replaces it with the correct one. This proofreading reduces errors dramatically, allowing replication to proceed with astonishing accuracy. It is as though the enzyme writes each letter of a manuscript while simultaneously rereading it, ensuring that mistakes are caught before they spread. But replication errors are only one source of damage. DNA is also vulnerable to environmental factors. Ultraviolet light from the sun, for example, can cause neighboring bases to bond inappropriately, forming distortions known as thymine dimers. Chemical agents, such as those found in smoke or pollution, can alter bases or create crosslinks between strands. Reactive molecules generated within the cell itself can break bonds or introduce modifications. Even the normal processes of metabolism produce byproducts that can harm DNA. To cope with this constant barrage, cells rely on specialized repair pathways. One such pathway is base excision repair. When a single base is altered or damaged, enzymes known as glycosylases detect the irregularity and remove the faulty base. The sugar phosphate backbone is then cut. The gap is filled with the correct base by DNA polymerase, and the strand is sealed by ligase. This process is like replacing a single misspelled word in an otherwise intact text, ensuring that the larger message remains coherent. Another pathway, nucleotide excision repair, is used for larger distortions, such as thymine dimers, caused by ultraviolet radiation. In this case, enzymes cut out an entire segment of the damaged strand, removing not just the faulty bases, but also the surrounding region. DNA polymerase then fills in the missing sequence, and ligase seals the strand. This approach is broader and more thorough, like cutting out a damaged page of a manuscript and rewriting it afresh. For breaks that occur in the DNA backbone, cells employ different strategies. Single-strand breaks can often be repaired using the undamaged complementary strand as a guide. Double-strand breaks, which are more serious, require more complex mechanisms. One option is non-homologous end joining, in which the broken ends are directly reconnected. This method is quick, but prone to small errors. Another option is homologous recombination, in which the cell uses a similar or identical DNA sequence, often from the sister chromatid, as a template for repair. This method is slower but highly accurate, restoring the sequence with precision. The choice between these pathways reflects the balance between speed and accuracy, between immediate survival and long-term fidelity. Mismatch repair provides yet another safeguard. After replication, even with proofreading, occasional errors may remain bases that are mismatched, but not structurally distorted. Enzymes scan the DNA, identify the newly synthesized strand, and remove the mismatched section. The gap is then filled with the correct sequence. This system is like a final review, a careful rereading of the manuscript after it has been copied, to catch any lingering mistakes. Together, these repair mechanisms create a network of protection that is both comprehensive and adaptable. They do not prevent damage from occurring, for such damage is inevitable in the dynamic environment of the cell, but they ensure that damage is corrected swiftly and efficiently. Without them, life would quickly unravel under the weight of accumulated errors. With them, life persists, generation after generation, with a fidelity that is nothing short of remarkable. The importance of DNA repair becomes even clearer when we consider what happens when these systems fail. Defects in repair pathways can lead to the accumulation of mutations, which, in turn, can give rise to diseases such as cancer. For example, individuals with inherited mutations in certain nucleotide excision repair genes develop a condition known as xeroderma pigmentosum, which makes them highly sensitive to sunlight and prone to skin cancers. This vulnerability underscores the protective role of repair systems and how essential they are for health. And yet, while the failure of repair can bring harm, the occasional imperfection it allows has also been a source of creativity and evolution. Mutations, though often harmful, sometimes confer advantages. They can produce new traits, new adaptations, new paths for survival. In this way, DNA repair strikes a balance. It prevents most errors to preserve stability, yet it allows just enough variation to provide the raw material for change. Life has endured not only because of repair, but also because of this delicate interplay between fidelity and flexibility. Reflecting on DNA repair, one is struck by its quiet persistence. It is not a dramatic or sudden process, but a continuous and patient one, unfolding within every cell, at every moment. It is a reminder that resilience is often not the absence of damage, but the ability to respond and adapt. Just as a community weathers storms by repairing what is broken, so too do cells preserve their integrity by constantly mending their DNA. To picture this is to see the cell not as a fragile entity, but as a resilient system, equipped with tools to meet the challenges of existence. Damage is inevitable, but destruction is not. Through repair, the instructions of life are preserved, copied, and carried forward, sustaining the continuity of species and the persistence of individuals. It is a quiet miracle of biology, a testament to the wisdom of nature's design. As we continue to explore the cell cycle, it is worth carrying this image of repair with us. The unseen caretakers, the patient editors, the watchful guardians of the genome. They remind us that life is not flawless, but it is enduring, sustained by the constant effort to mend what is broken. DNA repair is not only a mechanism of biology, but a principle of existence, one that ensures continuity in the face of inevitable change. 12. When things go wrong, cancer. For most of the time, the cell cycle unfolds with remarkable harmony. Cells grow, prepare, copy their DNA, divide, and rest, all under the quiet guidance of checkpoints and repair systems. The choreography is so well balanced that it is easy to take for granted, to imagine that it always unfolds without error. Yet in truth, the cycle is not infallible. When the safeguards falter, when the signals that govern growth and division are lost or ignored, cells may begin to divide uncontrollably. This loss of regulation is the essence of cancer, a condition not born of an external invader, but of the body's own cells, forgetting their balance. It is important to approach this subject gently. Cancer is often spoken of in terms of conflict or alarm, Yet within the framework of biology, it is simply a breakdown in the systems of control that normally preserve order. Cells are meant to divide only when needed, when tissues must grow, when wounds must heal, when old cells must be replaced. They are also meant to rest when their work is complete, or to step aside entirely when damage is too great. In cancer, these instructions are bypassed. A cell continues dividing even when division is unnecessary, producing more and more cells without pause. The cycle, once steady, becomes unrestrained. At the heart of this shift are changes to DNA. Mutations in certain genes can alter the signals that regulate the cycle. Some genes, known as proto-oncogenes, normally encourage cells to grow and divide when conditions are appropriate. When these genes are altered, they can become oncogenes, pushing the cell to divide without limit. Other genes, known as tumor suppressor genes, act as brakes on the cycle, stopping division when damage is detected. If these brakes are lost, cells can move forward unchecked. Still others are involved in DNA repair, and when they fail, mutations accumulate more rapidly, feeding the cycle of instability. In this way, cancer does not arise from a single mistake, but from the gradual layering of changes that erode the safeguards of the cell. It is striking to realize how many safeguards must be bypassed before cancer emerges. A single mutation is rarely enough. Instead, cancer is the outcome of many small disruptions, collected over time, each one weakening the cell's ability to regulate itself. This is why cancer is often associated with aging. The longer a cell line persists, the more opportunities there are for mutations to accumulate. Yet it is also why cancer remains relatively rare compared to the countless cell divisions that take place every day. Most errors are corrected, most damaged cells are removed, most divisions proceed without harm. Only in the unusual case where many layers of defense are breached does uncontrolled growth take root. When it does, the consequences unfold gradually. A small group of cells begins dividing more than it should, forming a cluster known as a tumor. Not all tumors are dangerous. Some remain benign, growing slowly, and remaining confined to one place. But when cells within a tumor acquire further changes that allow them to invade surrounding tissue or to spread to distant parts of the body, the tumor becomes malignant. In this state, the balance of the body is disrupted as tissues are crowded, signals are distorted, and normal functions are impaired. What makes cancer especially significant in the context of the cell cycle is how it reveals the importance of control. Mitosis, when regulated, is a source of healing and renewal. The same process, when unregulated, becomes harmful. It is not the division itself that is dangerous, but the loss of timing, the absence of pauses, the silence of checkpoints. Cancer is, in essence, a reminder of how much life depends not only on activity, but also on restraint. Even in this disruption, however, there is a kind of perspective to be gained. Cancer demonstrates that the systems of the body are deeply interdependent. A single mutation may alter one pathway, but it takes many changes across many systems for the full collapse of regulation to occur. The fact that these safeguards normally hold so well is a testament to their strength. The rarity of cancer compared to the vast number of cell divisions that occur every day is itself a quiet reassurance of the resilience of the cycle. From a broader view, cancer also highlights the role of variation in evolution. The same mechanisms that allow for diversity and adaptation, mutation, recombination, and change also carry risks. Life exists in a delicate balance between stability and variability, between fidelity and flexibility. Cancer arises when that balance tips too far, when the forces of change overwhelm the forces of stability. Yet without change, life would stagnate. The very processes that make us vulnerable to cancer are also those that have made us adaptable, creative, and capable of evolution. When we reflect on cancer in the context of sleep learning, it is helpful to hold it not as a fearful concept, but as an illustration of balance. It shows us that the cycle of division is not simply about growth, but about the harmony between growth and control. Checkpoints, repair systems, and programmed cell death exist to maintain this balance, and for the most part, they succeed. When they falter, we see the consequences, but those consequences remind us of the importance of the safeguards themselves. It is also important to note that cancer is not a single entity, but many different conditions. Each type arises in its own way, from its own combination of mutations and circumstances. Some progress slowly, others quickly. Some remain localized, others spread. Each reflects a unique story of how control was lost. Yet in all cases, the underlying principle is the same: the disruption of the cell cycle's careful regulation. And so, in our exploration of the cell cycle, cancer serves as a contrast, a demonstration of what happens when balance is broken. Mitosis, without regulation, is no longer renewal, but excess. Growth without pause becomes burden rather than strength. Yet this very contrast deepens our appreciation for the normal cycle, for the way it usually unfolds with such quiet reliability. The resilience of life lies not in the absence of threats, but in the strength of its systems to prevent them. As we move forward, we will consider how cells, when functioning properly, use control not only to avoid harm, but also to ensure harmony. We will look at the significance of regulated division, the ways in which growth is guided and restrained, and the importance of balance for the health of organisms. But before that, it is worth pausing with the thought of cancer as a reminder that even the most remarkable systems can falter, and that life persists because it has built layers of protection to hold disruption at bay. 13. The significance of controlled division. Cell division, when considered simply as a biological mechanism, might appear to be nothing more than a way for organisms to increase their number of cells. But beneath the surface of this process lies a deeper truth. Division is only valuable when it is controlled. Just as a flowing river can bring life when guided within its banks, but destruction when it floods unchecked, so too is cell division beneficial only when carefully regulated. The significance of controlled division lies not in the act itself, but in the balance. It provides the balance between growth and restraint, between renewal and stability, between continuity and change. Every living organism depends on this control. From the earliest moments of development, when a single fertilized egg divides again and again to form the first tissues, the timing and order of division must be exquisitely regulated. Cells divide to create the layers of an embryo, to shape organs, to form systems that will sustain life, but they do not divide randomly. Signals guide them, instructing some to multiply rapidly, others to pause, and still others to specialize. Without this guidance, the body would not take form. The very structure of an organism is built not only on division but on the restraint of division, on knowing when to multiply and when to stop. This principle continues throughout life. Controlled division allows tissues to renew themselves without overwhelming their neighbors. In the skin, for example, new cells are produced at the base, then slowly move upward as older cells are shed. If division were too slow, wounds would remain unhealed and protective layers would thin. If division were too fast, the skin would thicken uncontrollably, disrupting its function. In the bone marrow, blood cells are produced in vast numbers each day, yet their production is finely tuned to match the body's needs. Too few would leave the body vulnerable. Too many would crowd the system. It is the control of division that maintains harmony, ensuring that each tissue is balanced. The importance of control becomes even more evident when we recall what happens in its absence. Cancer, as explored previously, is essentially division without restraint. Cells multiply without responding to signals, ignoring the pauses that should guide them. The result is not renewal but burden, not healing but harm. This contrast highlights the quiet significance of regulation in the normal cycle. When division proceeds under control, it is one of life's greatest strengths. When control is lost, it becomes one of its greatest vulnerabilities. Controlled division is also vital for repair. When the body is injured, signals are sent to surrounding cells, instructing them to divide more rapidly to replace what has been lost. But once the wound is healed, those signals cease, and division slows again. This ability to respond with precision, to accelerate when needed, then to stop at the right moment illustrates the balance that makes healing possible. Without control, wounds would either remain open or produce excess tissue that disrupts the body's form. With control, the process is smooth, effective, and restorative. Another layer of significance lies in the way controlled division interacts with specialization. Cells do not simply divide to make identical copies. They also differentiate, taking on specialized roles as they multiply. The coordination between division and differentiation is essential. For example, in the nervous system, stem cells divide to produce both more stem cells and specialized neurons. But the number of neurons must be carefully regulated, for too many or too few would disrupt the delicate architecture of the brain. Controlled division ensures that tissues grow not just in number but in proportion, maintaining balance among the many types of cells that together form the body. The significance of control can also be seen in aging. Over time, many cells lose their ability to divide, either because their telomeres, the protective caps at the ends of chromosomes, have shortened, or because signals have instructed them to remain in a permanent resting state. This decline is not a failure, but another form of regulation, preventing damaged or unstable cells from dividing further. Though it contributes to the gradual slowing of renewal with age, it also protects the body from errors. Here again, the principle of balance is evident. Life is sustained not only by division, but by the careful limitation of division when it is no longer safe. Controlled division plays a central role in development across all forms of life. In plants, the growth of roots and shoots depends on regions of active division, known as meristems, where cells multiply under careful control to extend stems and leaves. In animals, embryonic development unfolds as waves of division and specialization, each guided by precise signals. In both cases, the elegance of form, the branching of a tree, the symmetry of a body, arises from division that is regulated, timed, and guided into patterns. Without control, growth would be shapeless and unsustainable. There is a poetic resonance in the idea that life's strength comes not from endless activity, but from regulated activity. The pauses, the checkpoints, the moments of restraint are as important as the divisions themselves. Just as music depends on silence between notes, and just as a heartbeat depends on its rhythm of contraction and relaxation, so too does the cell cycle depend on the balance between growth and pause. Controlled division is the rhythm that sustains life, preventing chaos and ensuring harmony. Reflecting on this, one can see why nature has built so many layers of regulation into the cycle. Growth signals, inhibitory signals, checkpoints, repair mechanisms, and even programmed cell death all work together to ensure that division remains under control. Each layer is a reminder of how vital regulation is. The cycle itself is not fragile. It is robust precisely because it is so carefully monitored. The persistence of life across billions of years rests on this resilience. Controlled division also provides the foundation for diversity. Because division is regulated, organisms can invest in long-lived, specialized cells that do not divide frequently, such as neurons and muscle fibers, while still maintaining tissues with rapidly dividing cells. This balance allows for complexity, for the coexistence of stability and renewal within a single body. It is the regulation of division that makes such complexity possible, allowing organisms to grow larger, live longer, and adapt more flexibly than they could otherwise. Ultimately, the significance of controlled division is that it transforms a simple Act 1 cell becoming 2 into a principle of harmony that sustains life itself. It is the quiet force that allows growth without chaos, renewal without excess, healing without harm. Without control, division is destructive. With control, it is creative, life-giving, and enduring. As we reflect on this, it is worth carrying the image of controlled division as a rhythm, steady and balanced, unfolding in silence within every living being. It is a rhythm that has sustained species for ages, a rhythm that shapes our bodies, repairs our wounds, and allows us to live in harmony with ourselves. The cell cycle is not merely about multiplication, but about regulation, about the balance between activity and rest. In that balance lies the persistence of life, the quiet assurance that renewal will continue, not recklessly, but with care. 14. Cell division across organisms. When we speak of the cell cycle, of mitosis and meiosis, of checkpoints and repair, it is often with human cells in mind. Yet the truth is that cell division is not confined to our own bodies, nor even to animals alone. It is a universal process, shared across the living world in many forms, each adapted to the needs of the organism. From the simplest bacteria to the tallest trees, from the tiniest insect to the vast expanse of a whale, the act of division sustains life. To reflect on cell division across organisms is to see not only the differences in how it unfolds, but also the deep unity that connects all living beings in this common rhythm. In the simplest organisms, prokaryotes such as bacteria, the process of division takes a form known as binary fission. Here, the cycle is far less complex than in eukaryotic cells, which possess nuclei and elaborate internal structures. In binary fission, the circular DNA molecule of the bacterium is replicated, and the two copies are attached to different regions of the cell membrane. As the cell grows, the membrane elongates, gradually pulling the DNA copies apart. Finally, the cell pinches inward and divides, creating two daughter cells, each with a complete copy of the genetic material. It is a process both direct and efficient, allowing bacteria to multiply rapidly. Indeed, under favorable conditions, some bacteria can divide every 20 minutes, producing vast populations in a matter of hours. This simple cycle sustains life in oceans, soils, and even within other organisms, reminding us that the foundations of division are ancient and enduring. In eukaryotic organisms, plants, animals, fungi, and protists, the cycle is more intricate, reflecting the complexity of the cells themselves. Here, DNA is housed within a nucleus. Chromosomes are organized into linear structures, and organelles must be distributed to daughter cells. Yet despite these differences, the underlying purpose remains the same: to preserve and pass on life's instructions. Whether in the tissues of an oak tree or in the lining of a human intestine, mitosis follows the same essential stages, guiding chromosomes from condensation to separation, from unity to renewal. The details may differ, but the rhythm is familiar, echoing across kingdoms of life. In plants, cell division carries a unique character. Because plant cells are surrounded by rigid cell walls, the process of cytokinesis differs from that in animals. Instead of pinching inward with a contractile ring, plant cells construct a new wall from within. Vesicles filled with cell wall materials gather at the center of the dividing cell, fusing to form a structure known as the cell plate. This plate expands outward until it merges with the existing cell wall, creating two separate but connected cells. The result is a structure that maintains the integrity of the plant's framework while allowing growth and renewal. In this way, Plants weave their tissues together, building stems, leaves, and roots through the quiet persistence of cell division. Animals, by contrast, rely on flexibility. Their cells are not bound by rigid walls, but by pliable membranes, which allows tissues to move, bend, and reshape. Cytokinesis here is accomplished by the tightening of actin filaments, pulling the membrane inward until it cleaves in two. This flexibility allows animals to develop a wide variety of tissue forms, from the soft expanse of muscle to the fine networks of nerves. The versatility of animal tissues reflects the adaptability of their cells, each one dividing in response to signals that guide development, repair, and renewal. Fungi, too, display their own adaptations. In yeasts, which are single-celled fungi, division occurs either through mitosis, followed by cytokinesis, or through budding, in which a small daughter cell grows directly from the parent before separating. In filamentous fungi, cells divide along elongated hyphae, sometimes with incomplete separation, leaving pores that allow materials to flow between neighboring compartments. These variations show how cell division adapts to different life strategies, shaping organisms for their ecological roles. Protists, the diverse group of eukaryotes that includes amoebae, algae, and many others, also reveal the adaptability of the cycle. Some undergo mitosis, much like plants and animals, while others employ unique methods of nuclear division. In certain algae, for example, nuclei may divide multiple times before the cell itself divides, producing many daughter nuclei that are then enclosed in separate cells. In ciliates, the presence of both a large macronucleus and a smaller micronucleus requires specialized forms of division, with each nucleus following its own cycle. These examples remind us that cell division, while universal, is not uniform. It has been shaped and reshaped by evolution to meet the needs of different ways of life. Across multicellular organisms, the context of division also varies. In some tissues, such as the growing tips of plant roots or the lining of animal intestines, cells divide rapidly and continuously, providing renewal where it is most needed. In other tissues, such as the nervous system, cells divide rarely, if at all, relying instead on longevity and stability. This diversity of division reflects the balance between growth and specialization, between renewal and preservation. It is a balance that allows complex organisms to function as unified wholes, each part sustained by the rhythm of the cell cycle. What unites all these forms, bacterial fission, plant mitosis, animal cytokinesis, fungal budding, protist variations, is the principle of continuity. In every case, the purpose is the same: to preserve the instructions of life and to pass them on to the next generation of cells. The strategies may differ, but the rhythm is shared. Each organism, no matter how simple or complex, depends on the act of division for survival and renewal. It is soothing to think of this universality. When you look at a tree, its growth is driven by cells dividing in its meristems. When you watch a flower open, its petals have been formed by countless rounds of division. When you consider the healing of a wound on your skin, it is the same rhythm at work. Even the microscopic life drifting in a drop of pond water depends on division to persist. Across the vast diversity of forms, cell division is a common thread, weaving together all living beings into the same fabric of existence. The significance of this perspective lies in its reminder of unity. Life on Earth is astonishingly varied, yet beneath that variety lies a shared mechanism, a rhythm that has endured for billions of years. Each bacterium dividing in the soil, each plant cell forming a new wall, each animal cell pinching into two, all are participating in the same ancient cycle. To reflect on this is to feel a sense of kinship with all living things, a recognition that we are part of the same continuity. As we continue our exploration of the cell cycle, We will turn from the diversity of organisms to the significance of division in growth and development. We will see how controlled division shapes the form of an individual, guiding the transformation from a single cell into a complex being. But before moving forward, it is worth pausing with the thought of cell division across organisms, of the shared rhythm that connects bacteria, plants, animals, fungi, and protists. It is a quiet reminder that beneath the surface of difference, life is bound together by the same enduring cycle of renewal. 15. The role in growth and development. When we look at the living world, one of its most striking features is the way in which it changes with time. A seed becomes a sapling, then a tree. A child becomes an adult. An egg divides into many cells, which organize into tissues, organs, and eventually a complete being. Underlying all of these transformations is the same quiet rhythm of the cell cycle, repeated again and again, guiding growth and development. While in earlier reflections we considered cell division as a means of maintenance and repair, here we see it in its creative role, shaping form and complexity from the smallest beginnings. The story begins with a single cell. In animals, this is the zygote, formed by the fusion of two gametes during fertilization. In plants, it is the fertilized ovule, resting quietly in the ovary of a flower. At first, this single cell seems small and unremarkable, but within it lies the complete set of instructions for building a living organism. The first act after fertilization is not to rest, but to divide. One cell becomes two become four, and soon a cluster of cells forms. Each round of division is rapid, with little time for growth between them, creating a mass of cells that will later specialize. This early flurry of mitosis is the foundation upon which all further development is built. As development proceeds, division does not cease, but its character changes. Cells begin to slow their pace, to grow between divisions, to follow signals that guide them into specific roles. Some cells become the lining of the gut, others form muscles, still others contribute to the nervous system. Division supplies the raw material, while specialization shapes that material into the intricate architecture of a living being. The coordination between division and differentiation is one of the most elegant aspects of development. Without division, there would be no increase in number. Without differentiation, there would be no form. Together, they create the patterns of life. In human development, this process continues for years. As a child grows, mitosis fuels the expansion of tissues, adding height to bones, mass to muscles, and resilience to organs. The same process shapes the leaves of plants as they unfurl, the branching of trees as they reach toward the sky, the growth of roots as they spread into the soil. Growth is not an event but a sustained rhythm, carried out by countless rounds of cell division, each one building upon the last. Controlled division also ensures that growth proceeds in proportion. A body is not a random collection of cells, but an organized structure in which each part develops in harmony with the others. This harmony arises from the way division is guided by signals. Growth factors, hormones, and environmental cues all influence when and where cells divide. In plants, light, gravity, and touch can shape patterns of division, guiding stems upward or roots downward. In animals, signals from neighboring tissues tell cells when to multiply, when to pause, and when to specialize. This communication ensures that growth produces balanced forms, limbs of equal length, leaves of symmetrical arrangement, tissues that fit together in seamless function. The role of division in growth is not limited to early development. Even after an organism reaches maturity, cell division continues to support expansion and adaptation. Trees add new rings each year, expanding in girth through the quiet multiplication of cells in their cambium. Muscles thicken with use, partly through cell enlargement and partly through new cell contributions. Skin grows and stretches to accommodate change. Life is never truly static, and division remains the quiet engine of adaptation throughout. One of the most remarkable aspects of cell division in development is its ability to generate complexity from simplicity. From a single fertilized cell, entire systems emerge. The intricate networks of neurons in the brain, the elaborate vascular channels that carry blood or sap, the fine structures of feathers, scales, or petals. This transformation is possible because division multiplies cells while allowing them to take on new roles. Each round of mitosis not only increases number but also provides opportunities for new instructions to be followed, new paths to be taken. The repetition of this process creates not mere masses of cells, but living structures with form and function. It is also worth reflecting on how cell division contributes to scale. A bacterium may be content to remain a single cell, but multicellular organisms achieve size and complexity through division. The human body contains trillions of cells, all descended from that first zygote. A giant redwood tree towers hundreds of feet above the ground, its trunk and branches built from billions upon billions of dividing cells. The sheer scale of these organisms would be impossible without the repeated cycle of growth. Division makes the small into the large, the simple into the complex, the invisible into the visible. Controlled division in development is also deeply connected to timing. Different tissues grow at different rates, some more quickly, others more slowly, depending on the needs of the organism. In human development, for example, brain growth is rapid in early years, supported by intense cell division, while other tissues, such as bones, continue to expand well into adolescence. In plants, leaves may form quickly during spring, while woody tissues expand more slowly over the course of the year. This timing ensures that growth is balanced not only in form but also in pace, allowing organisms to adapt to their environments and life stages. The role of division in growth and development carries lessons beyond biology. It reminds us that complexity arises not from sudden leaps, but from patient repetition. Each cell division is small in scale, yet together they build the vast forms of life. Growth is not a single act but a long process, shaped by countless small steps, each one contributing to the whole. In this sense, the rhythm of the cell cycle mirrors the rhythm of existence itself, where great transformations are built from many quiet increments. To pause and reflect on this is to appreciate the hidden labor within every living thing. The towering tree in the forest owes its form to centuries of divisions in its cambium. The newborn child, with delicate features and intricate systems, is the result of countless divisions since fertilization. Even the simplest flower, opening its petals to the sun, is the product of cell divisions guided with precision. Growth and development are not mysteries but the natural outcome of this rhythm, repeated faithfully across generations. As we continue to explore the cell cycle, we will turn to the role of division in repair, the way it restores balance when life is disrupted. Yet before moving onward, it is soothing to rest with the image of growth. A seed unfolding, a body lengthening, a being becoming itself through countless silent acts of division. The role of cell division in growth and development is not only to build but to guide, to shape, and to sustain, transforming a single cell into the complexity and beauty of life. 16. Division and repair. Throughout life, as organisms move through their days and years, the need for cell division does not end once growth is complete. The body is not a static structure, carved in stone and unchanging, but a living system in constant motion. Cells age, wear out, and are lost. Tissues are injured by accident or strain. Environments bring challenges that demand renewal. To meet these demands, cell division continues, not for expansion, but for repair and maintenance. This quiet rhythm of replacement sustains life throughout its span, ensuring that organisms remain whole and functional, even as time and circumstance leave their marks. The simplest form of repair is the regular turnover of cells. Many tissues in the body are in a constant state of renewal, as old cells are shed and new ones take their place. The skin is a familiar example. Each day, countless skin cells at the surface are lost, flaking away invisibly. Beneath them, in the basal layers, new cells are produced by mitosis. These young cells gradually move upward, maturing as they go, until they too reach the surface and are shed. In this way, the skin maintains its protective barrier, not by permanence, but by constant renewal. The lining of the digestive tract provides another example. Cells in the stomach and intestines are exposed daily to food, acids, and enzymes. Their lifespan is short, often only a few days, before they are sloughed off. Yet the tissue does not thin or weaken, because new cells are continually generated from stem cells nestled in the lining. These stem cells divide steadily, replenishing the tissue and preserving the body's ability to absorb nutrients. Without this ongoing cycle of division, digestion itself would falter. Blood is likewise maintained by division. Red blood cells, which carry oxygen, live for only a few months before they are broken down and replaced. White blood cells, the guardians of immunity, may live for shorter times, depending on their type and function. All of these cells arise from stem cells in the bone marrow, which divide to produce new blood cells every day. The scale of this renewal is vast. Millions of cells are created each second to sustain circulation, defense, and balance. The body depends on this silent, ceaseless production, as essential as breath itself. Beyond routine renewal, division plays a central role in healing. When the skin is cut, cells near the wound begin to divide rapidly, filling the gap with new tissue. Fibroblasts produce collagen to knit the area, while epithelial cells spread across the surface to restore the barrier. Over time, the wound closes, leaving behind a scar that marks the site of repair. A similar process occurs when bones break. Cells in the bone marrow and surrounding tissue divide to form a callus of new material, gradually strengthening and reshaping the bone until it regains its function. In muscles, too, small tears from exertion stimulate cells to divide, repairing damage and often making the muscle stronger than before. This capacity for repair extends even to more subtle injuries, those that occur at the cellular level. DNA, proteins, and membranes are constantly exposed to stress, and when they are damaged, cells respond not only with repair mechanisms, but also with replacement. If a cell is too damaged to function, it may undergo programmed death, and neighboring cells divide to take its place. In this way, tissues maintain their integrity, trading damaged parts for new ones, and ensuring that the organism as a whole remains strong. The ability of cells to repair tissues is not unlimited. Some tissues divide readily and heal quickly, such as skin and liver. Others, like nervous tissue, divide rarely, and their repair is limited. Yet even here, division plays a role. In certain regions of the brain, stem cells persist, producing new neurons that integrate into circuits. Though the process is slow and modest compared to other tissues, it demonstrates that the principle of renewal extends even to the most specialized parts of the body. In plants, too, division supports repair. A branch that is broken, a leaf that is damaged, or a root that is cut can stimulate cells to divide, producing new tissues that restore function. Plants rely on meristems, regions of active division that respond to injury by producing replacement structures. This capacity for regeneration is particularly striking in some species, which can regrow entire stems, roots, or leaves after damage. For plants, as for animals, division is the quiet mechanism by which life persists through injury. The significance of repair through division lies not only in its practicality, but also in its symbolism. It reminds us that resilience is not the absence of harm, but the ability to respond. Life is not preserved by avoiding all injury, but by meeting injury with renewal. Just as a forest regrows after fire, or a river reshapes its banks after flood, so too does the body restore itself through the rhythm of cell division. The act of repair is a quiet affirmation that damage does not mean an end, but a beginning of restoration. This process also reveals the balance between urgency and control. When tissues are damaged, cells accelerate their division to replace what is lost. But once the repair is complete, signals call them to pause, preventing excess growth. Too little division leaves wounds unhealed. Too much produces scars or imbalances. The harmony of repair lies in this balance. A surge of renewal followed by restraint, growth matched to need, restoration without excess. The persistence of division throughout life ensures not only healing but also stability. Without constant renewal, tissues would wither, organs would fail, and life would be short. With renewal, organisms endure, not unchanged, but continually refreshed. Each breath, each heartbeat, each step depends on cells that have been replaced through this hidden cycle. In this way, repair through division is not an occasional event, but a constant undercurrent flowing silently beneath the surface of life. To reflect on this is to see the body not as a fixed structure but as a dynamic process. What seems solid, the skin, the bones, the blood, is in truth in motion, rebuilt cell by cell through the ongoing rhythm of division. The person you are today contains countless cells that were not present months or years ago, cells produced by division to replace those that were lost. Identity endures, but the body renews itself continually, a living testament to the power of repair. As we move forward in our exploration, we will see how division also shapes the process of aging, how renewal slows and balance shifts with time. But before turning to that theme, it is worth pausing with the thought of repair. Cell division is not only the builder of bodies and the architect of growth, but also the healer, the restorer, the quiet caretaker of life. It is the rhythm that allows us to endure, to mend, and to carry forward, even in the face of wear and injury. 17. Division and aging. The story of cell division is often told in terms of growth and renewal, of beginnings and repair. Yet, like all rhythms in life, it is also shaped by the passage of time. As organisms age, the cycle of division does not disappear, but it changes. Some cells slow their pace, others stop altogether, and the balance between renewal and loss gradually shifts. This slowing, this gentle winding down of the cycle, is one of the underlying features of aging. To understand it is to see how the rhythm of division not only sustains life in youth but also guides the journey of life across its entire span. At the heart of this process lies the simple fact that cells cannot divide indefinitely. Each round of division brings with it a small but important change in the structure of DNA. At the ends of chromosomes are protective regions called telomeres, repeating sequences of DNA that act like the plastic tips of shoelaces, preventing fraying. With every division, these telomeres shorten, for the machinery of replication cannot fully copy the very ends of DNA strands. Eventually, the telomeres become too short to protect the chromosome, and the cell senses this as damage. At that point, the cell ceases to divide, entering a permanent resting state known as senescence. Senescence is not death. Senescent cells remain alive, carrying out many of their normal functions, but they no longer participate in the cycle of renewal. In this way, telomere shortening serves as a natural clock, limiting the number of times a cell can divide. This limit is sometimes called the Hayflick limit, after the researcher who first observed it. While some cells, such as stem cells and germ cells, possess enzymes that can rebuild telomeres and extend their lifespan, most ordinary body cells do not. The shortening of telomeres thus contributes to the gradual decline of renewal with age. The accumulation of senescent cells is only part of the story. Over time, damage also accumulates in DNA, proteins, and other cellular structures. Repair systems work diligently, but they are not perfect. Errors that slip through checkpoints, small injuries from the environment, and byproducts of metabolism all leave their marks. When cells are young, division helps to dilute these damages, as new cells are created with fresher components. But as division slows with age, this renewal becomes less effective. Damaged cells linger, repair grows less efficient, and the balance shifts toward decline. This slowing is not uniform across all tissues. Some parts of the body, such as skin and blood, continue to divide actively, even in later years, though at a reduced pace. Others, like neurons in the brain or muscle fibers in the heart, rarely divide after development, relying on long-lived cells to carry out their functions. In such tissues, aging is felt less as a slowing of division and more as the gradual wear of cells that cannot be replaced. Together, these differences shape the varied experiences of aging across the body. Thinning skin, slower healing, weaker muscles, diminished senses. Yet even here, the rhythm of division remains central. Stem cells, though fewer in number with age, continue to provide renewal. Blood stem cells still create new red and white cells, though at a slower rate. Muscle stem cells remain poised to respond to injury, even if their capacity is reduced. Neural stem cells in specific regions of the brain continue to produce new neurons, supporting memory and learning into later life. The persistence of these cells is a quiet reminder that aging does not end the cycle, but merely changes its pace. The role of division in aging also carries a kind of paradox. On the one hand, the decline in division contributes to the frailty of age. Tissues repair more slowly, wounds linger, and renewal is incomplete. On the other hand, the slowing of division also serves as a safeguard. If cells were to divide without limit, mutations would accumulate unchecked, and the risk of cancer would rise dramatically. Senescence, though it contributes to aging, also protects the body by preventing damaged cells from multiplying. In this sense, aging itself is partly the result of the body's efforts to preserve balance, a trade-off between renewal and protection. There is a poetic quality to this balance. Just as the seasons move from spring to summer, autumn, and winter, so too does the rhythm of division shift with time. Youth is marked by rapid growth, adulthood by steady renewal, and age by gradual slowing. None of these stages is less a part of life than the others. Each reflects a phase in the same cycle, a rhythm that guides organisms through beginnings, endurance, and eventual decline. When we reflect on division and aging, it is easy to focus on loss, on the slowing of renewal, and the accumulation of damage. Yet it is also worth considering the persistence that remains. Even in old age, cells continue to divide. Wounds may heal more slowly, but they still heal. Blood continues to circulate, renewed by marrow. The lining of the gut continues to replace itself, sustaining digestion. The body may be less resilient, but it is still maintained by the same quiet rhythm that carried it through youth. Aging also carries lessons about the importance of balance in the cycle. Too little division leaves tissues fragile. Too much division, without control, gives rise to disease. The gradual slowing with age is neither failure nor flaw, but part of the natural rhythm of life, shaped by evolution to balance renewal with stability. It ensures that life endures long enough to grow, reproduce, and contribute to the continuity of the species. It reminds us that mortality is not a defect of biology, but a part of its design. In plants, too, division slows with age. Young shoots and roots are full of dividing cells in their meristems, driving rapid growth. As plants mature, these regions remain active but less vigorous. Older trees may produce fewer leaves or flowers, yet they still grow new rings of wood each year. Even the longest-lived trees, standing for thousands of years, continue to depend on the rhythm of division, slowed, but never entirely absent. To contemplate division and aging is to see life as a continuum, shaped not only by beginnings but also by endings, not only by growth but also by decline. The cycle is not broken by aging, it merely changes tempo. The same rhythm that builds and repairs also slows and yields, guiding organisms gently toward completion. It is a reminder that life's strength lies not in permanence, but in the balance between renewal and rest, between growth and letting go. As we near the close of our journey through the cell cycle, we will next consider the broader perspectives: how the cycle unites all organisms, how it reflects patterns in nature, and how it embodies the enduring continuity of life. But before moving forward, it is worth pausing with the image of division and aging, the steady slowing of a rhythm, the gentle fading of renewal, the quiet persistence that endures even as time passes. 18. The cycle as a universal pattern. As we have followed the journey of the cell through growth, replication, division, repair, and aging, a wider truth begins to emerge. The cell cycle is not only a biological rhythm but also a reflection of a universal pattern found throughout nature. In its balance of beginnings and endings, of activity and pause, of continuity and change, it echoes the cycles that shape rivers and seasons, stars and galaxies, lives and generations. To contemplate the cell cycle as more than a cellular process is to glimpse the deep order that connects all things, the quiet symmetry of renewal that underlies existence itself. At its heart, the cell cycle is a pattern of repetition. A cell grows, prepares, divides, and rests, then begins again. This repetition is not meaningless, but purposeful, ensuring that life is sustained. In much the same way, the earth turns through its days and nights, its seasons of growth and rest, repeating the same cycles year after year. The repetition of sunrise is not monotony but assurance, the rhythm that makes life possible. The repetition of division within cells carries the same quality. Each cycle is both familiar and necessary. Each return to interphase is both continuity and preparation. The cycle is also a pattern of transformation. At each stage, the cell is not merely repeating itself but changing, moving from growth to synthesis, from condensation to separation, from renewal to rest. Transformation within repetition is a hallmark of cycles in nature. The tide that returns each day is never exactly the same, shaped by winds and moons. The seasons that circle through the years bring new growth each spring, but never in quite the same way. So too with the cell cycle. Each division creates something new, a daughter cell that is similar to its parent, yet distinct in experience and potential. The balance between continuity and change is perhaps the most striking feature of the cycle. DNA replication preserves the genetic instructions with remarkable fidelity, ensuring continuity across generations. Yet small variations, mutations, recombinations introduce change, allowing evolution to unfold. In this way, the cell cycle embodies the principle that life endures not by remaining static, but by balancing preservation with adaptation. This principle is reflected on larger scales as well. Rivers carve the same valleys, yet alter their paths. Forests regrow after fire, yet with new compositions. Stars burn steadily, yet evolve toward different fates. Continuity and change are not opposites, but partners in the unfolding of cycles. Another layer of universality lies in the role of pauses. Within the cell cycle, checkpoints serve as moments of rest and reflection, ensuring that conditions are right before moving forward. These pauses are not interruptions but integral parts of the rhythm, providing balance and safety. The same principle is seen in nature at large. Winter is a pause in the growth of plants, a time of rest that prepares for the vigor of spring. Night is a pause in the activity of day, offering renewal through sleep. Even the breath follows this pattern, with moments of stillness between inhalation and exhalation. The presence of pauses within cycles reminds us that rest is as important as movement, silence as vital as sound. The cycle also reflects a principle of universality across scales. The same rhythm that unfolds within a single microscopic cell is echoed in organisms, ecosystems, and even the cosmos. The renewal of tissues mirrors the renewal of forests. The decline of cells with age mirrors the decline of stars as they burn through their fuel. The death of individual cells, through programmed pathways, sustains the life of the larger organism, just as the death of stars seeds the universe with elements for new stars and planets. To see the cell cycle as part of this larger tapestry is to recognize the unity of patterns that connect the smallest with the vastest. In its quiet repetition, the cell cycle also speaks to the principle of resilience. Damage occurs, mistakes are made, but repair systems restore balance. This resilience is mirrored in ecosystems that recover after storms, in species that adapt to changing climates, in communities that rebuild after disruption. The cycle teaches that resilience is not the absence of harm but the capacity for renewal. Life persists not because it is unbroken, but because it can be mended. There is a calm assurance in recognizing the cell cycle as part of a universal pattern. It tells us that life is not a straight line but a rhythm, not an unbroken march but a series of returns and departures. Growth leads to division, which leads to renewal, which leads again to growth. The rhythm is circular, not linear, and in that circle lies continuity. This perspective is soothing, for it suggests that life is never truly lost, but transformed, carried forward in new forms, sustained by the same cycle repeated through time. In human experience, these patterns find resonance. We grow, rest, work, and heal, following rhythms that echo the cell cycle. Our lives move through stages of youth, maturity, and age, reflecting the stages of cellular renewal. Even in memory and tradition, there is continuity and change. Stories passed on with fidelity, yet altered slightly by each retelling. The cycles within cells are mirrored in the cycles of human existence, reminding us that we are not separate from nature but participants in its patterns. To reflect on the cycle as a universal pattern is to feel a sense of connection. The smallest cell, dividing in silence, participates in the same rhythm as the largest structures of the universe. Each is part of the same principle of renewal, the same balance of continuity and change, the same interplay of movement and pause. This connection brings a sense of unity, a recognition that life is woven from repeating rhythms that transcend scale and form. As we come toward the conclusion of our journey through the cell cycle, we carry with us not only the details of mitosis, meiosis, checkpoints, and repair, but also this broader perspective that the cycle of division is part of a universal pattern. It is the rhythm of life itself, echoed in seasons, tides, stars, and stories. A reminder that existence is not a straight line, but a circle of renewal. 19. The cycle as a universal pattern. Throughout this journey, we have moved carefully through the steps of the cell cycle, from interphase with its quiet preparations, to the choreography of mitosis, to the special role of meiosis, and to the safeguards of checkpoints and repair. We have looked at the ways division supports growth, healing, and the continuation of life, and we have reflected on how it changes with aging. Yet when one steps back from these details, something broader comes into view. The cell cycle is not simply a biological mechanism, but an expression of a deeper rhythm, a universal pattern of renewal that is echoed throughout nature. In its balance of growth and pause, preservation and change, beginnings and endings, the cycle resonates with the larger order of the world around us. At its most fundamental, the cell cycle is a rhythm of repetition. One cell becomes two, two become four, and on it goes. This repetition is not wasteful, but sustaining. It ensures continuity, allowing organisms to live, grow, and repair. The same quality can be found in the cycles that shape the natural world. The earth turns, bringing night and day, seasons circle, bringing growth in spring, abundance in summer, rest in autumn, and quiet in winter. Rivers flow to the sea, only to return again in clouds and rain. The repetition of the cell cycle is of the same kind, dependable, quiet, and life-giving. It reminds us that progress is not always a straight line, but often a circle, where each return to the beginning is also a continuation. Yet within this repetition, there is transformation. The cell does not merely repeat itself identically, but changes with each cycle. DNA is copied, yes, but small variations sometimes arise. Daughter cells may differentiate, taking on new roles. Through meiosis, entirely new combinations of genetic material are created. In this way, the cell cycle preserves what is essential, while also introducing change. This interplay of continuity and variation is mirrored in larger cycles as well. Each spring may resemble the last, yet each brings new blossoms, new weather, new life. Each tide may return, but the sands it shapes are never quite the same. Life is sustained not by sameness alone, but by the weaving together of preservation and transformation. Pauses are another essential feature of the cycle, and they too reflect universal patterns. Within the cell cycle, checkpoints act as moments of stillness, pauses where the cell waits until conditions are right. These pauses may feel like delays, but they are in fact essential, ensuring balance and preventing harm. The same is true in the rhythms of the natural world. Winter is a pause in growth, a time of rest that prepares the land for spring. Night is a pause in the day, a time of quiet that restores energy. Even our own breathing follows this pattern, with moments of stillness between inhalation and exhalation. Pauses are not interruptions, but parts of the rhythm, balancing movement with rest. The universality of the cycle is also evident when one considers scale. At the smallest level, a single cell divides in silence, unseen and unnoticed. At the largest level, galaxies themselves move through cycles of formation and dissolution. Stars are born, live, and eventually fade, seeding the cosmos with material for new stars. Between these extremes, ecosystems follow cycles of growth, disturbance, and recovery. Species emerge, flourish, and give way to others. The same principles of repetition, transformation, and pause are present at every scale, from the microscopic to the cosmic. To reflect on the cell cycle is therefore to glimpse a pattern that is not confined to biology, but woven into the fabric of existence itself. Resilience is another theme that connects the cell cycle with universal patterns. Cells encounter damage constantly, errors in replication, breaks in DNA, stresses from the environment, yet they repair, correct, and continue. This resilience allows life to persist despite inevitable harm. The same is true in nature at large. Forests regrow after fires, rivers find new courses after floods, communities rebuild after storms. Resilience does not mean that harm never comes, but that recovery is possible. The cell cycle, with its capacity for repair and renewal, reflects this same principle of resilience that sustains life in all its forms. There is a soothing assurance in recognizing these patterns. It tells us that life is not a series of disconnected events, but part of a greater rhythm. Cells grow and divide, not once but endlessly, each cycle linked to the next. Organisms live and age, but their offspring carry forward the rhythm. Seasons turn, tides return, stars rise and fall. In every case, the rhythm is circular, not linear, and within that circle lies continuity. Even endings are not final, but part of a larger cycle of renewal. For humans, these patterns find a special resonance. We too live in cycles of waking and sleeping, of work and rest, of childhood, maturity, and age. Our communities have cycles of growth and change. Our traditions repeat across generations. Our own lives echo the larger patterns of nature. The cell cycle, unfolding quietly within us, is both a literal mechanism of our biology and a metaphor for the rhythms that shape our experience. It is a reminder that we are not separate from the natural world, but participants in its patterns. As we rest with this thought, it becomes clear that the cell cycle is more than a subject of study. It is a reflection of existence itself, of the way life endures by balancing preservation and change, activity and rest, growth and decline. Its universality offers comfort, suggesting that beneath the complexity of life lies a simple, enduring rhythm. To contemplate the cycle is to feel connected, not only to one's own body, but to the whole of nature, to the earth, and even to the cosmos. And so, as this reflection draws to a close, we see the cell cycle not merely as a series of molecular steps, but as a universal pattern of renewal. In its repetition, transformation, pauses, resilience, and continuity, it mirrors the greater rhythms that shape all things. To hold this image is to find calm assurance that life persists, not in straight lines, but in circles, not in permanence, but in renewal, not in isolation, but in unity with the whole. The cycle is both the smallest rhythm within us and the widest rhythm beyond us, a gentle reminder of life's continuity through time. 20. Final synthesis and gentle closure. As we come to the final part of this long exploration of the cell cycle, it is time to gather together all that has been considered, to weave the many threads into a single fabric, and to let the story settle into a place of calm. We have traveled from the smallest details of DNA replication to the largest perspectives of life's universality. We have looked closely at mitosis and meiosis, at checkpoints and repair, at growth, healing, and aging. We have reflected on what happens when balance is lost, and why control is vital. And through it all, we have seen how the cycle sustains life with a rhythm that is both practical and profound. This final reflection is not a summary of details, but a gentle closing, a moment to appreciate the whole pattern as one continuous movement of renewal. The cell cycle is, at its essence, a rhythm of life. It begins in quiet preparation, builds toward division, resolves into renewal, and returns again to rest. Each cycle is a complete story, yet none stands alone. Every cycle leads to the next, just as every breath leads to another, every day to another, every season to another. This endless repetition is not monotony but assurance, the gentle promise that life continues. To rest in the thought of the cycle is to rest in the knowledge of continuity, of a rhythm that has endured for billions of years and will endure for as long as life persists. What makes the cycle so remarkable is its balance. It holds together opposite qualities in harmony. It preserves the genetic instructions with extraordinary fidelity, yet it allows for variation and change. It drives growth with energy, yet it pauses with patience at checkpoints. It supports resilience through repair, yet it also accepts decline with aging. It creates, sustains, and lets go, never clinging too tightly to one stage, always moving gently into the next. This balance is what allows life to flourish, not in rigid permanence, but in flexible continuity. The cell cycle also teaches us that renewal does not require drama. It unfolds quietly, invisibly, within each cell. There is no sound, no spectacle, only the silent pairing of bases, the careful alignment of chromosomes, the soft pulling apart of chromatids. Yet from these hidden movements arise the greatest of transformations. A body grows, A wound heals. A species continues. An ecosystem endures. The power of the cycle lies not in noise or force, but in its quiet persistence. This offers a lesson in stillness, that the most profound changes are often the most silent, and that continuity is sustained not by sudden leaps, but by steady rhythms. Another lesson of the cycle is that life is not a straight path but a circle. In each cycle of division, the cell does not march forward towards some final destination, but returns to a beginning that is also an ending. Interphase leads to mitosis. Mitosis leads back to interphase. Growth leads to rest. Rest to renewal. This circularity is mirrored in so many patterns of existence. The rising and setting of the sun, the turning of the seasons, the ebb and flow of tides, the birth and passing of generations. The circle reassures us that endings are not final, but openings into new beginnings, that life is always being carried forward in new forms. As we reflect on this circle, it is worth pausing to imagine the countless cells dividing within your own body at this very moment, in your skin, in your blood, in the lining of your stomach. Cells are quietly renewing you, sustaining your life without your awareness. Around you, in plants and animals, in soils and seas, the same cycles are unfolding. Bacteria dividing, roots extending, leaves forming, embryos developing, all following the same rhythm, each part of the same great continuity. To feel this is to sense connection, a kinship with all living things, bound together by the same cycle of renewal. Even in decline, the cycle carries meaning. With age, division slows, repair becomes less efficient, and balance shifts. Yet this too is part of the rhythm. Just as autumn is as essential as spring, just as night is as essential as day, so too is the slowing of renewal part of life's pattern. It is not failure, but fulfillment, a gentle winding down of the same cycle that once built and repaired. In this way, the cycle teaches acceptance, showing that every phase has its place, every stage, its purpose, every moment, its value. To see the cell cycle as both specific and universal is to hold a dual perspective. On one hand, it is precise and molecular, with enzymes and proteins guiding each step. On the other, it is symbolic and expansive, reflecting patterns that shape not only cells but the whole of life. This duality is not a contradiction, but a harmony. The small details make the large patterns possible, and the large patterns give meaning to the small details. In this sense, the cell cycle is both the hidden mechanism within us and the great rhythm beyond us. In these closing moments, the cycle offers us one final image, that of continuity. Life does not persist because any one cell or organism endures forever, but because the cycle repeats. Each cell passes its instructions to the next, each generation to the next, each species to the next. Continuity lies not in permanence but in renewal. This is the quiet assurance of the cycle, the gentle truth that life carries on, always changing, yet always connected, always moving, yet always returning. And so, as we let this reflection settle, we can hold the cell cycle not only as a subject of biology, but as a source of calm. It reminds us of resilience, patience, balance, and unity. It connects us to the smallest details of our own cells and to the widest patterns of the universe. It assures us that life is not fragile chaos but enduring rhythm, not broken line but continuous circle. To rest with this image is to rest with the thought of renewal itself, the gentle cycle that has always been and will always be.